Good afternoon, everyone.
Afternoon, everyone. Welcome to day 2.
Welcome, Dan. Thanks for having me today. It's a pleasure to be here.
No, thank you. It's after yesterday's first day, we had some really interesting talks about the first 12 months of diagnosis. Today, we're going to be focusing on the science and research, and I think Beer is going to give a brief intro to what we're going to be discussing today. So, Beer, I hand it over to you.
Thanks, Dan. Good afternoon to everyone. I am Beatriz Fernández, founder of ACTAYS, the Spanish charity for Tay-Sachs and Sandhoff and other neurological diseases, based in Madrid. And I'm very glad to be able to join today our annual conference together with the rest of the European charity colleagues. I want to inform to our families, first of all, that this conference will be uploaded in YouTube within a few days. So you will have the chance to see it with subtitles. So welcome to this second day of meetings and knowledge sharing. We are going to have some of the researchers that are going on some interesting scientific approach. After a first day talking about the first year after diagnosis and caring and clinical experiences, today we are going to discuss about some research projects going on about potential treatments. It is important for us to clarify that this conference is a forum of discussion, but not about announcements. We are not talking here about the start of clinical trials or about treatment available. So we would like to highlight that if you are looking for information about trials, it is important that you refer to your main doctor or medical team. So this said, I want to welcome our first speaker, Dan, that will give a 12 months review on the last months on the research into GM2 and how he has worked with industry to support their work. So Dan, everything for you. Thank you very much.
Thank you, Beer. Thank you. So the first talk we wanted to share with everyone today was really focusing on the research that's been undertaken for GM2. You're going to be hearing a lot about that over the coming hours from the different companies who are involved. But also we wanted to touch on our involvement within advocacy and also with our community to show how important we are in actually driving a lot of this work forward. Because without our voice, and I say our voice as the patients and the community, a lot of this research would not be able to move as far forward as it has in such a short space of time. So one thing that I think is very important for our community to know is why we are important and why we're important into research, why we're important for the companies looking at developing treatments. The really important thing that we can do as a patient community is that we can represent ourselves. And I think that's very, very important, that we can give an honest appraisal of when we're looking at different drugs being developed and how it can impact our community and our families. Why? Because we know the diseases, and that's something that all of us have become experts in. I'm sure many of you would have been in the same situation my family was in and Beer's family was in, is when your family member is diagnosed with Tay-Sachs and Sandhoff, you're told you will become the expert. Well, if we are being told that we will become the expert about these diseases, we need to be listened to. And I have to say, we're in a very fortunate position that many of the companies that we have worked with really appreciate and value the view that we can give. And we give an honest, upfront view of the work they're doing because we know the impact of these diseases on our family members. I think that is how our realistic view can really shape how a lot of these clinical trials are being developed and designed. We can't forget that anything that's being developed has to make sure it meets the needs of our community. And to do that, our voice must be heard. And as I mentioned previously, we are very fortunate that many of the companies we work with and many of the clinicians associated with those studies really listen to the patient voice when they're putting these together. When we look at the trial development, we know that there are key elements to the jigsaw, and many of you will have seen this slide before, and I think it's very important to keep highlighting this back to our community. Research is vital. We need to work with the researchers looking at treatments, looking at developing novel drugs to see how they can impact GM2. And we do that by working with those teams all across Europe to help provide the patient voice and drive that work forward. On top of that, we then work with our industry partners, and a lot of these companies can make a reality the research that's being done. They can help drive these things forward so they can go into the clinic and then potentially go to market. And that's something we've been spending a lot of time on the last few years in terms of our development as a patient group, is how we can make sure that we can work with industry partners with their work. And on top of that, we have the regulators and the patients. We work very closely with the regulators and with the patient community, as you know, to make sure that we can make these studies designed in a way that impacts our community. We basically need to make sure that when studies are being developed and when they are being launched, that actually they're going to meet the needs of our community. That not only does the trial and the drug work, but it actually has a real clinical benefit. And that's where we can step in the patient community. We can describe and explain what that actually is. And I think that's why we have to make sure that we're continually in these conversations as we're doing that. And this is where we sit. We're that missing piece in that jigsaw that can bring everyone together. We work directly with all these key stakeholders to make a reality happen. And I think that's a very important part for us as a community to really understand that our voice, our lived experience, what we go through on a day-to-day basis is absolutely vital when they're looking at developing treatments and to move them forward. We look here kind of, why is advocacy important? And many of you will be members of the different patient groups that are part of the ETSCC. Our role is to communicate with those stakeholders in a very, very clear manner. We make sure the information they have is shared. And when it's shared, it is information shared in a way that we can all understand. We touched on that yesterday, especially in the first 12 months of diagnosis, how information can be widely spread, how also it's in very technical language. What we do with our industry partners is to make sure that we can translate those documents down into something that we, the layperson, can really understand and understand their work and what it's doing. Something else that's really, really important within advocacy is we're always available. And many of the companies know they can always reach out to us for advice or to ask us questions. And our view is we need to make sure that we can support and drive these studies forward by being available to answer those questions and making sure the patient voice is heard, as I mentioned previously. And finally, and I think this is something that we are all very good at within the Tay-Sachs and Sandhoff community, none of us are ever afraid to give our view. And I think that's very important. We're always willing to support the companies and the researchers and the clinicians as they drive their work forward, but we make sure that our view is heard and our view is given. And without having that strong voice, we would not be in the position where we are today, where such an ultra-rare diseases such as Tay-Sachs and Sandhoff, having so many companies involved as they develop different treatments. Now, collaboration is something I'm a big believer on, and many of you will know this from previous talks, and this is something that ETSCC is very proud of as well, in that we work very closely together. We work with the international partners, both in the clinical side and the advocacy side, because we think it's vital that we come together, that we don't work in silos. And by actually doing this, we can make a much bigger difference as a community. And that's why I put together this slide. I think it's a really interesting one for us all to look how far we've come. When we go back to when the ETSCC was set up in 2012, there was in fact NTSAD, the Tay-Sachs Foundation, CATS and ACTAYS, and the Mathias Schmidt Foundation. All the other groups have started to come up and join our GM2 group. And you're going to hear later today about the global GM1 and GM2 alliance that's also going to be launched in 2024, with the goal really to bring us all together as an international global alliance and how we can work collaboratively and collectively to drive forward research. And again, this is another very, very important thing we need to do as a community. As more and more of our families are being diagnosed with these devastating diseases, we need to make sure they have somewhere to go, somewhere to go for support, somewhere to go for information. We want people to know there is a community out there they can become a member of, which can really support them as they go through this journey that many of us have been through. And I think it's very important also to recognise the organisations which have been working tirelessly in our space. Azafaros, Sanofi, JCR and Intrabio are doing lots and lots of work to actually provide potential treatments for our community. And this is something which you're going to hear more about as the talks go on. But it's very, very interesting to know that these companies are very active and working incredibly hard to drive their work on because they want to support our community. And they're doing something which we touched on yesterday, which again, I think is very important for us, is that they're giving us hope. The hope is that one thing that can get us through the darkest times. And when we think of that, I think back to when our daughter Amelie was diagnosed. We were told there was no hope for her. And we looked at hope in the sense of treatments, but actually a lot of the hope that we then were able to latch onto was support, hope of a community. And now we have that established, not just within our own countries, but actually on a European level. And then on a global level, when we look at the Global Alliance, it's actually going to make a much bigger difference to our community as we are then able to drive forward those treatments, working with our industry partners. So what I want us all to really reflect on and understand is that we are trying to work hard every single day to give hope back out to our community and back out to our families. And we are here to support them through the journey, both with family support, but also looking at it from a treatment side. And I look back when, Amelie was diagnosed, and then when she sadly passed away, we had hope with our community that we would be there for each other. And that's something I'm proud that we have been able to establish, not just within our countries, but also within Europe. Because our dream in 2032 is to be here. We want to have global studies available. We've set a very, very high target. That's something we're really committed and keen to do. We know that there's a lot of more hard work that has to be done. But I think as a community, we can really support everyone who's working towards that goal. The question is, how do we get there? Well, we get there by working collaboratively, as I mentioned before, not just within advocacy in our community, but also with the companies, the clinicians and the researchers there. We're never going to stop working for our community because we know what they're going through every single day. And that's something that motivates and drives us forward. And we always think about their families. They are all the people who inspire us to do our work every single day. They're the reason why we hold these events. They're the reason why we do the fundraisers. These are the reasons why we spend many hours working to drive forward these treatments. And that's something that I think is very, very important as we continue to advance forward the treatments for GM2. What we all want to see? Well, we want to stop people passing away from these really cruel diseases. We want people to see that we are a collaborative group with the researchers doing their work. And they can also look at us as a standpoint as someone who can support them all the way through. And I think that's something we should all be very proud of. So we really want to say thank you for the dedication and the hard work, not just to our researchers, to the companies out there, but also to our community, because your dedication really helps us drive forward the work that we're doing. And we're very grateful for everything you've done in supporting us as we've gone through our mission to make sure that we can continue to support the Tay-Sachs and Sandhoff community, not just with their support from the families, but also the support within research. So thank you very much. I'm going to stop sharing my slides and I'm just going to get our next speaker online. So I will be back very shortly. I'm just going to put up a very quick placeholder screen. In fact, I'm going to play a very short video instead about what is Tay-Sachs and Sandhoff, and we will be back shortly.
If someone you care for is affected by Tay-Sachs or Sandhoff disease, you may be wondering what this means. Keep watching the video to find out more. What are Tay-Sachs and Sandhoff disease? Tay-Sachs and Sandhoff disease are part of a group of diseases called GM2 Gangliosidoses. They are rare inherited diseases that affect the brain. There are three main variants of the diseases which are classified by the age of onset. These are infantile, juvenile, and late onset. A condition is considered rare if it affects less than 1 in 2,000 people. There are approximately 300 million people worldwide impacted by rare diseases. Everyone has an instruction manual needed for their body to work. These instructions are known as genes and are passed on from parents to child. Tay-Sachs and Sandhoff disease are caused when a child has been passed on 2 copies of a certain gene that has changes or mutations. They inherit 1 copy from each parent. Parents can be a carrier for these mutated genes without being affected by the disease. These genetic changes are not something that can be prevented. What happens inside the brain in Tay-Sachs and Sandhoff disease? In someone not affected by Tay-Sachs or Sandhoff, the cells of the brain work like a factory. The genes instruct how many workers there needs to be and what their jobs are. When the factory is working efficiently, it produces a molecule called GM2, which helps the brain work efficiently. Once GM2 has done its job, it's then sorted and removed by a group of factory workers called Hex A. However, in people with Tay-Sachs or Sandhoff disease, the mutated genes mean there aren't enough Hex A workers. This means GM2 can't be removed properly, and it starts to build up, until the factory, the brain, is unable to work as well as it should. This leads to problems with thinking and speaking. It also causes problems with movement of the body, such as standing, walking, and eating. Common symptoms of Tay-Sachs and Sandhoff disease may include fits or seizures, muscle weakness and loss of muscle tone, particularly affecting the chest and stomach area, increased jumpiness to unexpected noises, reduced coordination, delay reaching development milestones such as sitting or walking, difficulty with speech, inability to eat unassisted, and difficulties with thinking and learning. Tay-Sachs and Sandhoff disease are progressive and multi-system conditions. This means that in time, more parts of the body and bodily functions will be affected, and the person will need more assistance. Unfortunately, these conditions also lead to a limited life expectancy. Whilst there is currently no cure, treatment for Tay-Sachs and Sandhoff disease focuses on helping parents and their families to manage and ease the effects of the diseases. The healthcare team involved in supporting people with these conditions and their families may include specialist neurologists, physiotherapists, and dietitians, amongst others. At first, a diagnosis may be overwhelming and can make those impacted and their families feel isolated. But although Tay-Sachs and Sandhoff disease are considered rare, there is a strong community providing support to families, such as the CATS Foundation. It may take time to adjust to living with Tay-Sachs and Sandhoff disease, but eventually these changes can become a new normal. Together, families and friends can support one another and provide those affected by GM2 Gangliosidosis with the best possible life.
So there's a slight change in the agenda. We're now going to have Taylor Fields from Intrabio, who's going to be presenting the work that they've been doing. So sadly, Taylor can't be here in person, but we have a recording that she did for a presentation, which I will be sharing in 1 second. And we'll just play now.
Everyone, my name's Taylor Fields. I'm the Chief Product Development Officer and Senior Vice President of Intrabio, and I'm delighted to be presenting remotely and via video today for the 10th Annual European Tay-Sachs and Sandhoff Family Conference. I'm going to be giving an update on our development of N-acetyl L-leucine, or IB1001, for the treatment of GM2 Gangliosidosis. N-acetyl-leucine is a chemically modified amino acid that can be formulated as 3 distinct drugs. N-acetyl-D-L-leucine, a 50-50 mix of the stereoisomers N-acetyl-L-leucine N-acetyl D-leucine. The best analogy I have to describe this is if you look at your left and right hands and hold them individually, you know that these do different things, and if you were to cross them, they would do different things. This is the way these three compounds work in the body. They are metabolized differently, and they have different pharmacological effects. N-acetyl D-leucine had been initially approved in France in the 1950s for the treatment of acute vertigo. It goes by the trade name Tanganil and has not been approved anywhere else in the world for any other indication. Our team in parallel has spent the last 13 years working on this compound to identify that the L enantiomer, N-acetyl L-leucine, is the active enantiomer with a superior safety and efficacy profile when it's administered independently. It's very well tolerated, it's taken orally, and we have ample preclinical and clinical evidence that N-acetyl L-leucine has a symptomatic and neuroprotective disease-modifying effect for various disorders, including GM2 Gangliosidosis. As I mentioned, N-acetyl L-leucine, or IB1001, is an orally administered agent. We know that N-acetylation is a huge aspect of its pharmacological activity, and that without that chemical modification, L-leucine alone would have no therapeutic effect. So the drug is taken orally as granules in a sachet, and this new novel transport mechanism enabled by the acetylation process allows it to be delivered to all major organs as well as across the blood-brain barrier to the brain. Here, we know that the drug has polypharmacological effects, including the normalization of mitochondria function, so producing more cellular energy, the normalization of lysosomal function, and has knock-on effects which include the dampening of neuroinflammation and the stabilization of neuronal membrane potential. All of these together accumulate to have these rapid symptomatic benefits, but also offer the long-term neuroprotective effects which I described. What I'm going to discuss today are the results of the three most recent studies conducted with IB1001, namely the phase 2b studies conducted for both GM2 Gangliosidosis and the related lysosomal storage disorder Niemann-Pick disease type C, which used an identical phase 2b trial design, and the most recent phase 3 pivotal study conducted with IB1001 for NPC. The phase 2b studies with NPC and GM2 used a master protocol, meaning that they were two separate studies with identical study designs. The goal of these studies were to demonstrate that IB1001 could rapidly improve symptoms after 6 weeks of treatment, followed by a 6-week post-treatment washout. The studies were conducted at multinational centers across the US, the UK, and Europe, and enrolled patients 6 years and older with a confirmed diagnosis of either GM2 or NPC. The drug was orally administered, and the studies were ran as open-label studies, meaning that the investigators and the patients and their families knew when they were receiving IB1001. However, the primary endpoint was designed to be based on videos taken of each patient at each visit and assessed by independent blinded raters who had no notion of when patients were on treatment or off treatment, and then could therefore make unbiased and informed assessments of the true treatment effect. The results of the primary and secondary endpoints from the 201 NPC and 202 GM2 are presented here. In both studies, IB1001 met its primary blinded endpoint, but also its secondary endpoint, the scale for assessment and rating of ataxia, and clinical global impression of change assessed by the parents, caregivers, patients, and investigators. The changes which you see in blue are the changes during the treatment period, i.e. the improvements while on drug, while the boxes in orange are the changes during the post-treatment washout, where patients essentially return to baseline. We were very pleased that there was internal consistency between the primary and secondary endpoints in each trial, but also that the results of these two parallel studies in two parallel diseases were identical, which really reinforced for us the treatment effect. The major conclusions from both of these phase 2b studies were that IB1001 was an effective treatment for the neurological signs and symptoms of GM2 and NPC. So in both trials, the drug met its primary and secondary endpoint, and there was a rapid improvement in symptoms in 6 weeks. The clinical relevance of this was patients' improvement in walking, gait, balance, fine motor skill, speech, swallowing, and cognition. So functional abilities that were hugely relevant to patients' everyday life. The drug was well tolerated in both studies with no treatment-related serious adverse events, and in both studies we saw this rapid improvement during the treatment period, followed by a deterioration or return to baseline during that post-treatment washout period. Across all subgroups, regardless of the patient's age, their age of disease onset, or their severity at the beginning of the trial, there was an improvement, reinforcing the belief that this should be a treatment for all patients with GM2 and NPC. And the data were directly consistent with the preclinical and prior observational studies we had conducted for GM2 and NPC. We've completed now the analysis of the extension phase, which is unpublished, but will be shared shortly. And in both studies, we saw, after 2 years, a stabilization of disease progression and a neuroprotective disease modifying effect. After both of these studies were completed, we very candidly had hoped that the data would be used to support a new drug application for the FDA. Unfortunately, in our discussion with the agency, they confirmed to us without placebo data, we would be unable to file an application. The NPC study had completed about 6 months before the GM2 trial, so we were a little bit more ahead with this program, and we decided to go forward and run the placebo-controlled trial that the FDA wanted in order to secure approval for the drug. So we designed a multinational, placebo-controlled, randomized crossover study that looked at patients during a baseline period, after 12 weeks of treatment with either IB-1001 or placebo, and then they would cross over to receive 12 weeks of treatment with the opposite therapy, which meant that patients received both IB-1001 and placebo during the study and were able to be used as their own control. The goal of the study was to again demonstrate that IB-1001 was an effective treatment for the neurological signs and symptoms of NPC, and we used very similar endpoints to the phase 2 study, including the scale for assessment and rating of ataxia, the clinical global impression scale, the modified disability rating scale, and quality of life questionnaires. We were very pleased that we completed recruitment for this study in under 3 months, and while we had wanted to recruit 46 patients, we actually surpassed that total and recruited a total of 60 patients. In July of this year, we shared the very exciting announcement of the results of this placebo-controlled trial, and directly consistent with the phase 2b studies, IB-1001 showed a statistically significant and clinically meaningful improvement versus placebo on all of its endpoints. So it met both its primary SARA, but also all secondary endpoints. The clinical effects of which were quantified through various exit interviews and clinical global impression scores, where we saw that the treatment benefits, as seen in phase 2, were hugely meaningful and relevant to patients' everyday life. As in the phase 2 studies, the safety profile of IB-1001 was again demonstrated to be very well tolerated and benign. There was an equal number of adverse events that occurred between the treatment and placebo groups. There were no adverse events with IB-1001 with over 10% of patients experienced, and there were no serious adverse reactions. As the next steps in our program, we're currently very active with finalizing the new drug application to be submitted to the US FDA for IB-1001 for the treatment of NPC. It is scheduled for this to be submitted in January of 2024. In parallel, we're working with the EMA and plan to submit the marketing authorization application in April 2024 for IB-1001 for NPC. We have requested and confirmed a meeting with European regulators in February 2024 to discuss the fastest route for drug approval for IB-1001 for GM2 Gangliosidosis. We are fully committed to getting IB-1001 approved as a treatment for Tay-Sachs and Sandhoff in both Europe, the UK, and the US, and want to do everything that we can to leverage the successful placebo data from the NPC study to hopefully support a quicker timeline for approval for GM2. So in the coming months, I will absolutely keep the community updated. I'm sorry I don't have more concrete pathways at this time, but wanted to reinforce and reassure that we remain committed to getting IB-1001 approved for GM2 and are working to do that as quickly as possible. Thank you again for the opportunity to present today. I've listed my contact details here in case there are any follow-up questions. I've also listed the link to the clinicaltrial.gov page of the GM2 study, where you can find the complete study protocol as well as the parent study results, and the link to the publication of the parent study GM2 Gangliosidosis phase 2 results. Have a great rest of your meeting, and I look forward to having exciting updates for you soon.
So that was the update from Intrabio. So Taylor messaged me earlier to say if anyone has any questions or needs more information, please either to reach out to myself or to Taylor directly, and we can coordinate a response to you. So it's exciting to have Intrabio obviously presenting again. They first came to our conference in, I think it was 2017, 2018. So to see the development of their work over the last few years is really exciting, especially when we look at the fact that this was started from scratch. And it's very exciting for us to have been part of that journey from the very beginning with that organization. So our next speaker, I'm very excited to bring in, is Mathias Schmidt. Hi, Mathias, how are you?
I'm doing well. Thank you so much. Thank you for inviting me.
No, thank you for joining us. Really excited to hear more about the work that JCR Pharmaceuticals is doing. So I'll leave you to introduce yourself. I'm going to put your slides up on to this stage, and you just let me know when you want me to flick through and just say next slide. That's fantastic.
Oh, yes, thank you so much. Then, so again, it's a big privilege to be at the conference. My name is Mathias Schmidt from JCR Pharmaceuticals, located in San Diego. So last year, we presented already at the CATS Family Conference. Next slide, please. This is our typical disclaimer. Don't believe anything that I say. Next slide, please. We will also talk about a drug that is only approved for MPS II in Japan. That is our disclaimer. Next slide, please. Now, who is JCR Pharmaceuticals? We are, for the most part, a Japanese company. Our headquarters is near Kobe. Our production and research is also in Kobe, but we have some subsidiaries in the US, Brazil, and Europe. Next slide, please. What's really important here is we have a little bit more than 900 employees, but most of them are really dedicated to research and development, 34%, and a big part to manufacturing. So we also try to manufacture, well, we do manufacture all our existing drugs on the market and experimental drugs ourselves. Next slide, please. Why do I believe we are a little bit different? Because for us, it really, what matters most for us, why are we in this field and why do we do what we are doing? So what are we doing? Yes, we're developing drugs for medicines with rare diseases. How are we doing it? Of course, we always have the highest sense of purpose, but only when we come to the why, our great sense of purpose that we receive from the parents and the patients, all the inspiration that we receive, and find it as a privilege really to work with all of you to fulfill some of your hopes and dreams. And I know sometimes they're not really big. It's like this. So I think everybody of you knows what GM2 Gangliosidoses is. You know exactly the pathology. There is a mutation of this enzyme called beta-hexosaminidase. It has an alpha and a beta subunit, and if they don't work, we have clinical manifestations on the right. And I think you are much, much more the expert than I am about the clinical manifestations. Next slide, please. Now, for many other lysosomal storage diseases, we have been exploiting this very deep understanding that we know exactly the pathophysiology, we know which enzyme is defect, just to manufacture the enzyme and to give it intravenously in order to treat those individuals affected with the disease. Now, the big question is, why does this not work for GM2 Gangliosidoses, for Tay-Sachs? Next slide, please. And the reason is, those enzymes, they're so big, they don't cross the blood-brain barrier. The blood-brain barrier is actually a wonderful invention from human nature to prevent the entry of noxious, toxic, or infectious agents into the brain, like viruses, bacteria, or anything that might be toxic to the brain. However, it also prevents the entry of enzymes. into the brain. Therefore, if you give the enzyme intravenously, it won't reach the brain. And GM2 gangliosidosis, to a very large extent, is a CNS-driven disease. Next slide, please. Now, people have really tried to bring this enzyme as close to the brain as possible. Companies have tried intrathecal delivery into the spinal fluid, but the problem is it only kind of surrounds it. And then there is an intraventricular delivery, which is slightly better, but still does not really solve the situation. The intravenous delivery would hypothetically the very best way to deliver drug to the brain, because the brain is highly vascularized, as you see on the right. And if it can leave the blood vessels, it would be the very best way of delivery. But for this, you need a platform technology to deliver it. Next slide, please. And what you see here is actually what JCR has developed is such a kind of shuttle technology to bring those enzymes across the blood-brain barrier. There is an antibody against a receptor on the blood-brain barrier that naturally transports larger molecules into the brain. And if we fuse an effector, like an enzyme, to this antibody, it binds to the very same receptor by a mechanism that we call receptor-mediated transcytosis. The enzyme is delivered actually not only into the somatic cells, but also into the brain. And this is not science fiction, because the first drug that was developed on this technology, it's called ITSCargo for the treatment of MPS II, was approved in Japan in 2021 and is marketed ever since. Next slide, please. Now, this drug is really postulated, all those drugs are postulated to work in the CNS, but also in the other cells outside of the body. Next slide, please. Next slide. So we have created such a molecule that has got hex-A enzyme and tested it in a mouse model where we knocked in the human transferrin receptor so that the antibody could bind. And we knocked out the hex-A enzyme so that the animals would resemble gangliosidosis. Next slide, please. Question is, does this molecule have any impact on the reduction of the substrate or on the function of the mouse? Next slide, please. What you see here is a previous study in those mice where we only looked at the substrate and the substrate reduction in certain tissues or organs of those mice. What you see on the very left is always the normal, the healthy mice. What you see on the next bar, the black one, is a strong increase in the substrate. And then you see treated animals either with the antibody enzyme fusion protein, JR479, or with the enzyme only. And what you can see is in the brain, spinal cord, cerebrospinal fluid, and actually also in the eye, you only see a reduction of the substrate upon treatment with JR479. But you don't see any reduction with the naked enzyme because the enzyme does not reach the brain, the spinal cord. It also does not reach the eye. So the hypothesis is really working. If you look into plasma or heart, where there is no such barrier, JR479 and the somatic enzyme only hexosaminidase A work actually equally well. Next slide, please. Now, the big question is, does it also have an impact? And what we tested here was gait, survival. I just show you the substrate concentration. So we started another experiment where we treated the animals when they were 11 weeks old at initiation of dosing. And we treated for 20 weeks and looked at the outcome. Next slide, please. So first of all, what we have observed that JR479 had a huge impact on the survival of the GM2G mice. So control mice would die around about 120 days, and we see a significant extension of life at both doses of JR479. Next slide. And then we also checked the gait, and there's a very sophisticated instrument. It's called the catwalk, where you can measure how mice go from A to B. And what you see on the right side, we looked how long it took the mice to reach the hole and what was the speed while they were getting into the hole. Again, you see the normal healthy mice and the diseased mice in the black bar. And you see a huge increase in duration, how long they take to reach the hole, and they're a lot slower. And then you can see a dose-dependent effect of JR479 to get them back to speed and to find them to allow them to find faster back into the hole. So we were very encouraged by the data that we have seen from this study. Next slide, please. Next, what we also did since last year, we checked whether the molecule reaches the brain of monkeys, which is very important. The monkeys are the very closest to humans, and the regulators, they require us to test this also in monkeys. Next slide, please. And what you see is the left side is not that important. Let's have a look at the right side. The drug concentration in the monkeys, in the brain, when we give either a low dose in pink or a high dose shown in red. And then you see various different areas of the brain. And what we can see is a very nice dose-dependent exposure of the molecule in the brain of the monkeys to a pretty nice extent, a significant extent where we believe this is therapeutically relevant. Next slide. So where are we right now? The manufacturing of those molecules is relatively complex. And we started the pilot manufacturing. There's a lot of process development associated with it. As we are speaking, we're doing safety studies that the regulators require us before we can go into clinical trials. And middle of next year, we are going to start GMP manufacturing under very controlled conditions, so that in the first half of 2025, we can start our clinical trials. Next slide. So where are we right now? We have this molecule, and this molecule is actually manufactured in very, very small cells from the ovaries of a Chinese hamster. It's actually cancer cells, they don't die, but it is very well established in the industry for many decades that these cells work perfectly for the manufacturing of recombinant biotherapeutics. And many, many approved biotherapeutics have been manufactured in CHO cells. They are then grown in a biofermenter of the size of somewhere between 2,000 and 2,500 liters. And you can see on the right the complexity of the biomanufacturing, but we're very confident that we can stay in the timelines that you have shown, that you have seen today. So we're very excited about this molecule. We're very excited that we are progressing according to plan, and we are looking forward to bringing this molecule into the clinic to hopefully alleviate some of the burden for the patients, and hopefully this molecule helps fulfill some of the hopes and dreams. I want to stop here and thank you very much for your attention. It's been a pleasure to speak with all of you. Thank you so much.
Sorry, I'm back on. Sorry, I was on mute. I say it's fantastic to have you present again and to see how far you're looking ahead and your development, and to see how far you've got already. And I think from our perspective as a patient community, it's encouraging to see that there is a really solid plan that you're working very, very tightly against. And obviously having a new kind of a new approach. We've seen small molecules, drug repurposing. So it's very, very exciting. I've got a comment here actually, if I can just put that onto the screen. I don't know if that's something you can answer. Can you comment on the cerebellum histology data? Is that something you can comment on? Would you want to take that offline?
So Ruben, I'm very sorry. There is so much echo in the line. It's almost impossible to, if you ask a question, I'm seeing that there was a question about the cerebellum Purkinje cells depletion histology data. There is no Purkinje cell depletion actually in the histology data. We're bringing the molecule into the Purkinje cells because they are very important. They have a very important function in the brain. So there is actually, this might have been a misunderstanding about depletion of Purkinje cells. They're still there.
Fantastic. And just obviously thank you from Diana from the NTSAD for a great talk. Super interesting. And I'm sure if anyone has any questions, they can reach directly out to you,
Yes.
and we'll share the details.
Feel free to share my email. Yes, of course.
Fantastic.
Thank you
Thanks,
so much.
Mathias. Really appreciate it. I'm going to get our next speaker up. So thank you. Have a good day. So our next speaker, very pleased to join us all the way from Germany, Dr. Mengel. Thank you. How are you?
Yeah, I'm fine. Thank you, Dan.
Fantastic.
Hello, everybody.
I'm going to share your slides. And I will leave it for you to introduce yourself and the presentation. And then, as mentioned before with the video, let me know when you want me to press play, and I'll press play.
No, I can move. Thank you. My name is Eugen Mengel. I'm from Germany, and I'm a physician related to lysosomal storage disorders for more than 25 years. And I have seen my first Tay-Sachs patient in 94, so that's nearly 30 years ago. And I like to report a little bit about our study in Germany. It's a natural history study. It describes the manifestation and the disease progression in gangliosidosis, and I'd like to start with we gave them a funny name. This study 8 in 1 stroke, and 8 in 1 stroke, it's a fairy tale from Germany, and it's a report about a tailor who creates the sentence 7 in 1 stroke, and he becomes a hero because 7 in 1 stroke, that's classic. That's super. And we thought, okay, the brave little tailor can 7 in 1 stroke, we can one more, 8 in 1 stroke. And 8 is a nice and important number. It reflects that we have here 8 gangliosidosis, and we use in the studies a scoring system. How severe is the disease? And this scoring system has 8 items. And moreover, the German word for 8 is acht. Sounds difficult for somebody who is not speaking German, but acht. And acht, you can play with this word, it had other meanings. For example, auf frühe Symptome achten means in English, watch for early symptoms. And den Patienten achten means esteem patients. So there are good reasons to call our study 8 in 1 stroke. And it's a multi-disease German natural history study. And this study is initiated not only from an investigator group, it's also investigated, and that's completely new in the field, from the patient advocacy group in Germany, hand in hand. And I give an update and report a little bit about the story of this study. So it's a new concept of clinical trials, patient and investigator-initiated trials, I like to call it. And it's a collaboration of the institute where I am, Sphinx, and a German patient advocacy group, Hand in Hand, or Hand in Hand in English. And we had already started this study in 22 and 2020, and we run in the first year exactly in the Corona area, and so it lasts more than 1 year that we and published it in 2020 our baseline study. So the data were created in 21 and published in 22. It's not so bad, but without Corona we would be earlier. And you see on the names on the publication, there are some investigators, clinical researchers, but also 2 members of the patient advocacy group, and they were an important researcher in this study. And some words to the 8 in 1 score. It summarizes 8 adaptive behaviors, I would say. It starts with participation and ends with cognition. And every item was rated from 0, which means no restrictions, and most were loss of function, rated with 4. And so a high score means a severe disease, and a low score means a low disease. And we have this done. Not only the investigators, the doctors have rated, also blinded the parents. And what you see here in this graph is the investigators on the x-axis and the caregivers on the epsilon axis, and the number and the rating. So there's a high agreement between the raters of the caregivers and the investigators. There is maybe some a little bit more, and that has to do with epilepsy in these children. So, and epilepsy in general, it's really a nightmare even for investigators to quantify epilepsy anyhow. And it's even more difficult seems to be more difficult for parents to quantify it. And we work on better explanations, but it's really difficult. And that's from the baseline data, my most loved graph. It shows every column shows 1 patient, and we developed a 6-phase defined 6 phases of the disease evolution. It starts with symptom-free and ends with minimal or no motor function, and in between the progression of the disease. And what's very clear here shown in the graph, that the greater the symptom-free in interval, the slower the disease progression. And second, what you see has some different clinical differences between GM1 and GM2. For example, here you see the late infantile GM2 patients and the late infantile GM1. And if you look to the colors, it's different. Starting with the symptom-free interval, it's somehow identical, but the progression rate is different. And what we also recognize in the newest data, very actually, that when we had more patients in the subgroups, that they are genetically different. The late infantile patients with GM2, they have no mild mutations, whereas in GM1 patients, the late infantile patients have a mild mutation. So we have to explore that more out, but there seems to be also a genetic difference next to it. What do we do? The study runs about 5 years. It starts with baseline. We had a medical history, a physical exam, and then most important, the 18-1 score. And to validate the 18-1 score, we used the Vineland Adaptive Behavior Score to look at and looked also to other important tests which are used in clinical trial. For example, Taylor reported about the Intrabio trial and the SARA and the 9-hole PEC4 test. They were done also in this trial, and so we like to create here natural history data to pass the way for new medications. And we also, and that's important for our newest update from the results, committed medications. I will say some word to that, and also to events, which means here mostly mortality. Yeah, the study enrollment, we started in the end of the first year in the trial with 26 patients, and here during Corona, we had about 10 patients included in the following year. And then in the next year until now, we had quite an impressive increase in the enrollment numbers. I think it has to do to Corona, and the second reason is we could, since some months, also include diseased patients, so the medical records of diseased patients, and the etiquette prove that, and that's important aspect and of the natural history. Yeah, 65 patients, the most of them, like in the most registries worldwide, are Tay-Sachs patients, and less patients with Sandhoff disease and GM1. And the other gangliosidosis are extremely rare, but however, two activator deficient patients, quite a number for this ultra rare disease. Maybe Sandhoff disease is more common in Arabic countries, I have the impression, but I have no exact numbers for that. Mortality, it shows that in infantile subtype, as we expected, but it's more quantifiable now that the infantile subtype die very early, and the first one, the first 5 kids here who passed away were GM1 patients, and then the GM2 patients followed, and the same in the late infantile patients, and also those patients who are still alive with late infantile subtype, they were, despite one, all GM1 patients. There is seems to be I'm very sorry. There seems to be a computer problem, or you do not see here the evolution of the 18-1 scores about 1 to 2 years. It's really a very difficult situation. We saw that it is increased, and I summarized 2 findings here, that that in the older patients, they start with a score to lose motor function, and with that they had a high increase, and then there was a plateau, and the second information here that we had with 1 to 2 years, but in the end we have to wait for 5 years. But now we saw in the infantile patients that the patients with epileptic seizures have a more rapid, and the other have in the final phase of their life a plateau effect. I'm sorry for that there is a mistake here. So, and what we have done more recently, it's that we had a closer look to juvenile and adult subtypes, especially those who were now adults, and we identified in the registry 16 patients they were adults, and they have a juvenile or adult subtype, and we looked for the 4 most important symptoms in GM2. And first one, dysarthria, and you see there is in all patients some impact of them with dysarthria. We have some impact, low motor neuron disease, so muscle atrophy and inability to move the legs and develop muscle strength there, and there are also, it seems to be a little bit correlated, dysarthria. The most severe patients with dysarthria had also the most severe lower motor neuron disease. And I highlighted the psychiatric symptoms. Not all adult patients had it, but some very severe, moderate, and some mild. But this is a significant number in impact in psychiatric disease, and that's our goal for 24, to study this in more detail, and may make a sub-study on psychiatric symptoms in these patients. And the cognitive deficits are not severe, especially in patients with adult onset. It's more severe in patients with juvenile onset, and they have more cognitive progression, I would suggest. And another issue we become aware is the use of cationic drugs in adults for psychiatric treatment. And the big question is, do cationic drugs lead to disease progression? And there is a basic science paper available from 2019 from the Konrad Sandhoff group, and they investigated the impact of cationic drugs on the catabolism of GM2 and found that they are even worsening the metabolic defect. And what's known also from the literature that more than 20 years ago, that GM2 patients often tolerate cationic drugs poorly. They have a lot of side effects with Parkinsonoid symptoms and severe dystonia. And what also known that not all, but some of the cationic drugs also interfere with Ganglistat. And we'll look here and see that from our 16 adult patients, more than half of them used cationic drugs, and made that some important information for patients in future who may like to use Ganglistat. It's necessary to think about that. So, in summary, the investigator and patient initiated a trial, 8-in-1 stroke, is running since more than 33 years, and 65, not 64, and 65 patients are enrolled. And I like to give an extra benefit from our 8-in-1 stroke trial, and and it's about an educational video about first symptoms in GM2. And we recognized as we take the medical history in our study that the first symptoms may are difficult to explain from parents, and they show us their handy videos where they filmed first symptoms, and it was for us very educational. And Birgit has the idea to create an educational video, and then may you can show it here, the video, or at least a part of the video here on this place.
Yeah, we'll play that now. So everyone, we're going to play a video, 5-10 minutes. You can see the resource that has been put together, and Dr. Mengel will come back on stage to discuss it. Here we go.
Hallo zusammen, ich bin die Tülay. Ich komme aus Nordrhein-Westfalen. Mein Sohn Ömer leidet an Tay-Sachs, ist 3 Jahre alt. Wir haben die Diagnose mit 17 Monaten erhalten.
Mein Name ist Melanie Mende. Mein Sohn Ben hatte infantiles Tay-Sachs. Die Diagnose haben wir kurz nach seinem 1. Geburtstag bekommen.
Mein Name ist Silvana Zilli. Mein Sohn Alessio ist 2019 an Tay-Sachs verstorben.
Ich bin Carmen. Ich bin die Mutter von Ava und Carla. Die sind beide von Tay-Sachs betroffen.
Mein Name ist Igor Katz, ich bin 52 Jahre alt. Ich bin unmittelbar betroffen, also nicht nur ich persönlich, meine Familie. Wir hatten ein Kind, das ist Sophie gewesen. Die Sophie hatte eine infantile Form der Tay-Sachs-Krankheit.
Also mein Name ist Heike Müller. Ich bin die Mama von der Melanie Müller. Sie leidet unter Tay-Sachs, der adulten Form.
Mein Name ist Yildiz Denitz. Ich bin die Mama von der kleinen Sivelia, die ist mittlerweile 7 Jahre alt geworden und ist leider an der schrecklichen Krankheit Tay-Sachs erkrankt.
Ja, also ich bin Nicole, ich bin 40 seit dem 22. September und ich habe seit 2018 die Diagnose Tay-Sachs.
Mein Name ist Birgit Hart.
Ja, und ich bin der Volker Quack und gemeinsam haben wir den Verein Hand in Hand gegründet.
Wir haben 2013 die Diagnose Morbus-Sandhoff für unseren Sohn Dario bekommen.
Mein Name ist Eugen Mengel, ich bin seit vielen Jahren Arzt, der sich mit lysosomalen Speichererkrankungen und unter anderem auch Tay-Sachs, Sandhoff und GM2-Aktivator-Defekt beschäftigt. Die Familien und Eltern, die machen Erfahrungen und nehmen Dinge wahr, die in der Apparatemedizin häufig untergehen.
Die ersten Symptome, die sind uns relativ früh aufgefallen. Die Schreckhaftigkeit, das war so das allererste, was uns sehr auffiel.
Fing es mit dem Erschreckhaftsein an. Das fing dann auch sehr früh an mit 4 Monaten, dass er extrem erschreckhaft war. Dass wir jedes Mal auch, wenn er geschlafen hat, ruhig sein muss, also Tür zu, kein Ton. Das war schon merkwürdig eigentlich, warum das so Extremfall ist.
Als dann eben die Krankheit fortgeschritten ist, hatte er eben diese Schreckanfälle. Also das heißt, dass er, also wenn er erschrocken ist, dann eben auch für kurze Zeit sein ganzer Körpertonus in sich zusammenbrach. Und er konnte zwar irgendwann damit umgehen, hat sich dann rechtzeitig festgehalten, aber das war dann schon so ein Zeichen, dass eben hier was sehr eindeutig auf die Krankheit hindeutet.
Und dann ist uns auch gefallen, also beziehungsweise unseren nächsten Freunden, dass Sophies Blick irgendwie ins Leere schielt. Ja, so. Und dann kamen wir auf die Idee, also, sie einem anderen Arzt vorzustellen. Und der hat gesagt, also, da stimmt was mit den Augen nicht bestimmt. Also, weil sie nicht die naheliegenden Objekte oder nahestehende Menschen erkennt oder Objekte. Und daraufhin sind wir dann zum Augenarzt, zum Professor in unserer Klinik in Karlsruhe. Und er hat bei diesem berüchtigten also kirschroten Fleck an den Augen, in den Augen entdeckt.
Dann war es so, dass mein Sohn mich nicht richtig angeguckt hat, wenn ich ihn ganz dicht auf dem Arm hatte und ich dachte, vielleicht sind es die Augen, vielleicht stagniert die Entwicklung deswegen. Und wir waren beim Augenarzt und der hat halt was auf der Netzhaut entdeckt. Und da ist der Kinderarzt dann aber auch sofort tätig geworden und hat uns dann wirklich ins entsprechende Krankenhaus überwiesen, dass wir eine genetische Untersuchung machen, weil dann auch bei ihm die Alarmglocken letztendlich losgegangen sind.
Sie kam verzögert also zu ihren Beweglichkeiten, also sie konnte sich auch mit 5 Monaten nicht alleine vom Rücken auf den Bauch drehen und wenn wir ihr dann ein bisschen nachgeholfen haben, war sie nicht in der Lage, ihren Kopf aufrecht zu erhalten.
Die ersten Symptome von Ömer, die gingen recht früh auch. Mit 4 Monaten hat es schon angefangen. Er hatte auch keine Kopfhaltung, also er hatte allgemein überall am ganzen, also keine er hat Muskelschwäche gehabt.
wurde es bewusst, so mit einem halben Jahr, 7, 8 Monaten, weil dann so Dinge fehlten wie, er konnte immer noch nicht sitzen, er fing nicht an zu krabbeln. Im Gegenteil, ich hatte das Gefühl, dass er auf dem Arm noch mehr in sich zusammensackte als vorher schon, also dass da sogar Rückschritte stattfanden.
Des Weiteren das Tragen. Also wir haben dieses Kind getragen und die war so schlapp und das war wirklich den ganzen Tag über.
Der Dario war von Anfang an sehr schlapp. In seinen Briefen stand dann drin, er ist hypoton. Er konnte im Grunde die Füße gar nicht oder die ganzen Beine gar nicht richtig bewegen. Er ist gerobbt nur auf den Händen. Und er hat sich sehr schwer getan, überhaupt zu laufen.
Also bei Melanie waren die ersten Symptome so ein bisschen Unsportlichkeit. Das war ungefähr so ab dem 7. Lebensjahr.
Also mit dem Laufen war es schon immer sehr schwierig und ich musste eben ausziehen, weil ich immer öfter die Treppe runtergefallen bin. Und dann Ja, also einfach mit dem Laufen. Also das Gangbild war schon immer auffällig und das Zittern. Also ich zittere, wenn ich trinke oder wenn ich esse.
Sonst war dann mit 3 Jahren bei der Ava von der Wechsel von Krippe in Kindergarten. Da hat sie angefangen mit Ängste-Situationen, Gleichgewichtsprobleme, Auffälligkeiten. 6 Monate später hat sie angefangen zu stottern. Die Sprache wäre immer undeutlicher.
Die verwaschene Sprache kam mit der Zahnspange, dass da schon so ein bisschen leichtes Genuschel war. Wir haben es halt auch einfach auf die Zahnspange.
So the video itself has another, I think about 10 minutes. I know we're going to be sharing that video into the chat and to the community later, but I know in the interest of time, Dr. Mengel, I'm going to put your slides back up for you to finish up. Here you go.
Yeah, I only like to summarize that this video illustrates first symptoms recognized by parents, and even the whole film is done with support from the parents. And the findings I explained in medical language for education, and it starts with symptoms in infants and followed by toddlers, school kids, adolescents, and in the end also adults. Here also the web link to the Hand in Hand website. Yeah, and I think that's my last slide. Here is the team of the Sphinx, and I still in memoriam of Michael Beck, who has in the beginning so much done for this study, and now he passed away in the last year. Sorry for that. Thanks a lot.
Thank you, Dr. Mengel. I think as everyone has said in the chat and in the sharing of the comments and everything, it's a great, fantastic resource that's been created by yourselves and Hand in Hand. And obviously the insights that you gave from the 18-month study, again, is absolutely fascinating. I know you shared the publication link. I think what we'll do, we'll put the publication into this chat as well so people can read it. And then if there's any further insights, please, it'd be fantastic then to share that with everyone here so we can really raise more awareness and understanding of GM1, GM2. Was absolutely fantastic.
Yeah, and maybe it was a good idea yesterday then to make about this publication also a non-medical language paper. Yeah, I think it's a great idea. We have to do that.
That's going to be, I think, when Kathy joins to talk about the Global Alliance idea we're having. That's one of the key deliverables. Will be producing resources that in a terminology that we can understand. That's fantastic. So thank you very much, Dr. Mengel. Again, if anyone wants to reach out to you, they can do so via the chat. And then I'm going to introduce our next speaker. So thank you very much.
Bye-bye.
So I'm very pleased to bring in Laura from Azafaros, who's going to give an update on the work that they've been doing. So Laura, fantastic to see you. Do you have your slides or would you like me to share them?
You can share it if you want.
Okay. As I'm doing that, please just introduce yourself and I'll get those slides up now.
Yes. First of all, thanks for inviting us to this conference, and thank you, Dr. Mengel, for changing the. I'm Laura Lopez. These are Gisela's slides from yesterday. I'm Laura Lopez. I'm the medical director at Azafaros for Europe. And as you all know, Azafaros is a pharmaceutical company working on the development of a drug for gangliosidosis. We are working on it. Let's see how it goes. We are hopeful with the drug. So let's see. I will start. We can move forward to the next one. This is a bit the agenda, and the next one it's just an introduction about the company. I will not extend too much because you know it. Just say that we are growing. Now we are about 25 employees, and we are most of us located around Europe. As you can imagine with my amazing accent, I'm not from Switzerland, I'm Spanish, and most of us are located in different countries. Next slide. Our compound, you know it from other meetings, it's Nibaglustat. It has a dual effect. It's an oral molecule with a dual effect. By one side, we inhibit GCS, which is the glucosylceramide synthetase. It's the same enzyme that it's inhibited by other substrate reduction therapies. And what we do in inhibiting this enzyme, it avoids to synthesize the molecules that will be stored. So in GM2 disease, we have a problem with the degradation of the GM2 molecule. So if we avoid the synthesis of this molecule, we cannot store it because we do not have it. This is what we do with the first effect, which is inhibit GCS. But we also inhibit another enzyme that is a non-lysosomal glucosylceramidase, the GGGA2. And the effect of inhibit this enzyme, it's not so much known. It's not so clear, but there are two different lines. By one side, we maintain the pH inside the lysosome. If you try to solve something in vinegar or in water, for sure the result will not be the same, because one liquid is more much acid than the other. The same happens in the lysosome. The lysosome needs an acid pH to work, but when a lot of things are stored inside, this pH starts to become more similar than water. What we are doing, inhibiting this enzyme, is help to regulate this pH, avoid that it becomes water, so the enzymes that are on it can work better. This by one side. By the other side, we also inhibit the synthesis of substance that in a human body, normal person, healthy person, do not have any impact, but that when they are stored, they become neurotoxic. As the brain of the patients have enough impairment because of the disease, we can avoid to storage other things that can even make worse. So we do not synthesize them. This is a bit the idea with the inhibition of the two different enzymes with the same compound. I hope you understood it. If not, please make me questions. Next slide, please. To make this drug and to put it in the market and be able to use it, we need to follow a clinical plan. It starts with the preclinical studies. We test the drug in animals. Then we move forward to a phase 1 in healthy volunteers, phase 2, and phase 3. At the same time as the phase 2, that is the one who is running right now, we are doing a couple of phase 1 studies to know more about the drug and to change the formulation. And simultaneously, we are working on a study that maybe it's the one most interesting for you right now, which is the Pronto study, an observational study for GM1 and GM2. And I will talk a bit more about this in the next slides. Please, Dan. Next one. Okay, this is the introduction of the Pronto study. It is a natural history study. We are not treating patients. We want to know how the disease evolved and which are the best assessments to see this evolution of the disease. To then can use this choose of these are the best assessments to design the phase 3 study for our compound. This study is running since 2022, and the idea is to have it until 2025. Next slide, please. We have 31 patients recruited in the study. Since the beginning, we activate 14 sites, but only 12 of them include patients with GM2 disease. We have 6 countries and 9 different nationalities included in the study, because we have the option to include patients fully remote, so patients who are not living in the country can perform the study there. In the timeline, you can see the recruitment period since May 22 to October 23, and the plan is then complete 2 years of follow-up of the patients in October 25. Obviously, we cannot be sure that this will be the real timeline because patients are not mandatory in the study. If there is a clinical trial that the patient can be included, if they are sick and they decide that it's too much, all the patients have that right to say, I'm stopping the study in any moment. This study and whatever. You can be in a clinical trial with a drug and you also can say, I'm stopping it. So we cannot ensure that the patients will arrive to the 2 years in October 25 or maybe sooner, depending on how the study evolves. Next slide, please. Next, okay. Here I say that we have 31 participants, but from now I will only talk about the GM2 patients. So there are 16 GM2 patients recruited in 4 countries with 4 different nationalities, and the distribution is not equal between Tay-Sachs and Sandhoff disease, as is the real life. In the previous video that we saw, most of the families are families with Tay-Sachs disease. So obviously in our study, we have much more Tay-Sachs disease than Sandhoff. Next one, please. This is a bit the distribution of the patients in the study, the time that they have ongoing. The first GM2 patient was included 19 months ago, so we are close to the 2 years of follow-up. Instead, the last patient included, it only had 3 months of follow-up. And I will not talk about the techniques that what are we measuring, because you already know to talk about this study, so I prefer present some results more than what are we doing. In the next slide, please, we can see some results regarding the baseline and the follow-up of the SARA. The SARA assesses the ataxia in the patients. The patients in the blue box are the ones who have 1 year of follow-up. And we can see that, for example, the baseline data regarding kinetics, on those patients, kinetics has a maximum score of 16. And we see that almost all the patients that have 1 year of follow-up are in the worst score for this disease. And we can see similar for gait posture, which the score is 18, they are a bit better, and for the speed. And this allows us to evaluate the progression, because we see that after 1 year of follow-up of these patients, in the speed we saw a worsening, in the gait posture we also saw a worsening, but if we do not know how is the baseline data in our patients, we can consider that the kinetics is stable. Yes, it's stable because it cannot be worse because they are in the higher score. That they cannot increase the score do not mean that they cannot be worsening. So we need to look for another way to evaluate the status of the kinetics on these patients. Next slide, please. We also have assessments not only in the motor part of the patients, also in the behavioral part. We have the baseline scale. At baseline, we saw that almost all the GM2 patients are in the low adaptive behavior. They have a scale that they adapt with the age. And most of the people are in this area here, which is what it's considered adequate, how you interact with the society, how you communicate, how you perform your daily life. This is where most of the people are, this area in between 85 and 120. Most of our patients have lower scores in all the capabilities, but mostly in the communication. And also when we evaluate the evolution, most of the patients worsening mostly in communication. They worsen in all the categories, but communication is more severe. They lose the capability to talk. Next slide, please. Regarding the phase 1, and now I'm moving forward to the clinical trials. The phase 1 was performed in 2021 in Australia with 35 healthy volunteers. Really important, healthy people, not patients. And we want to know if the compound is safe. Okay, so we are sure that we are not doing even worse the disease that the patients already have. Next slide, please. This study was designed in 2 parts. There was the part A, single ascent doses, and this means that we test our compound, 1 milligram of our compound, with a group of healthy population. It is safe, no new arms, no one lose the capacity to see each other, nothing worse happened, so we increase the dose to 3 milligrams. And we again evaluate that nothing bad happens, so we increase it. Once we arrive to the 9 milligrams and we know that it's tolerated, it's safe, we are not making harm to the patients, we move to the part B, which is a multiple ascending dose. Okay, take the drug 1 time in your life, it's not bad. What happens if you take it every day? This is what we do in the part B. And what we saw after the analysis of the data is that not severe adverse events appears, no gastrointestinal adverse events, which are common with other substrate reduction therapy, but we have mild and moderate skin-related events, none of them severe, and most of them treatable with a topical creams. So it's not a big issue, but it's not the perfect drug without any adverse event. These do not exist. Next one, please. Now, as we saw that there are not bad adverse events, that the healthy population can tolerate well the drug, we move forward to a phase 2 study with patients. It is running right now. The first idea was to run the study in US and Brazil, but with the status of the recruitment, we will close the recruitment only in Brazil. And we want to assess 2 things. First, if what we saw in the healthy population, that our drug is not bad, it's well tolerated, it's also real for patients. And the second thing that we want to assess is which is the best dose to treat our patients. Okay, next slide, please. And to do that, we designed the study with 6 patients with NPC and other lysosomal disease, 6 patients with GM2. We have a period of screening of 4 weeks. This means that since the patient is identified in the hospital to start the study, we have a frame of 4 weeks. If this frame is longer, if the patient needs more time to start the study, we have to reassess it. Because maybe the biochemical profile change, maybe it has an infection. There can be things that make the patient cannot be included in these 4 weeks. Then we have 3 months of the main study, where all the patients are randomized in the placebo or 2 different groups of Miglustat, 2 different doses. It's only 3 months. In 3 months, it's almost impossible. It's very difficult to find a clinical effect. So we are only assessing biochemical effects of our drug. We are checking which biochemical profile is better to treat our patients, but we do not expect miraculous recovery in 3 months. So it's important to know that, as you all know, it's a progressive disease, and in 3 months we can see a stabilization because of the disease do not progress just because of the natural disease of the history. After these 3 months, all the patients who are in the placebo group move forward to one of the to one of the two groups with drug, with treatment. So all the patients will receive treatment after 3 months of the study. And once we analyze all the data created in the main study and we know which is the best dose to treat the patients, all the patients will move forward to this dose. Okay, and they maintain it until our drug will be approved. Next slide, please. These are the inclusion and exclusion criteria of the study. We need the genetic confirmation of the diseases. We know that GM2 in several countries is diagnosed mainly by enzymatic activity, but we need the confirmation with the genetic result. Patients had to have some neurological disease because we are targeting the neurological symptoms of this disease with our drug. They must be older than 12 years old and able to comply the protocol. And then they cannot have other neurological diseases or not stable seizures. Patients have seizures, patients can have it. We cannot exclude those patients with seizures, but we need that every day or every week the seizures are more or less the same. Because if not, we cannot know if they are increasing seizures because of the drug. If they are reducing seizures because of the drug, they need a stabilization before I start the treatment. And more important for NPC than for GM, patients cannot be receiving Nibaglustat, which is approved to treat this other disease. Next slide, please. This is the current status of this study of the phase 2. We have 6 NPC patients included. We still do not complete the recruitment for GM2, but we are on it. And if nothing bad happens, next week we will have not 6, but 7 patients with GM2 included in the study. In the bottom part of the slide, you can see the timeline of the recruitment. We have 3 patients recruited before July, so they are now for sure in the extension phase where all the patients are receiving treatment. So if one of these 3 patients received placebo at the beginning, now for sure the 3 patients are under treatment and no one complains. This is the current status. The next slide, please. We also have, as you saw in the first slide, other studies. We have an ACTAYS study in healthy volunteers, and this comes because we expected much more drug. We had an expected way to remove the drug from our body, and what we thought that will happen, it's not what it's happening. So we can follow up with the phase 2. We know that it's not toxic, but we want to know how we process this drug. And this is what we are doing with the ACTAYS study. Know more about how this drug moves in our body since we take it until we eliminate it. We also have a bioavailability study that I will explain in the next slide. And we are preparing the phase 3 study that it's the one that we will use to prove that the drug is effective. Okay? The idea is to do a study worldwide, not as this phase 2 that it's not in Europe, for example. And I cannot talk about the inclusion-exclusion criteria of the number of patients we will have it because we are working on that. Regarding the bioavailability study, next slide, please, Daniel, and it's the last one. Currently, we are working with a capsule. We are working with a very small capsule. It's the size number 4. As you can see, it's smaller than 2 centimeters, and it's not commonly used to administer drugs. It's just for very special drugs. And in theory, size 2 is considered adequate for children and adults with swallowing problems. But what we realized with the study and what patients' organizations told to us is that it's not well received patients with a capsule, treatments with a capsule, because it's difficult to swallow for the patients. So we will move forward from the capsule to a more child-friendly formulation that will be easier to administer and that it also will be able to be administered via G-tube. But to do that, to do this change from the capsule to the new formulation, we need to compare two drugs. Our phase 1 and phase 2 data are with capsules, so we need to prove that it's exactly the same administered one form or the other. And when we demonstrate it's exactly the same, then we can do the change without problems. But this comparison, if I receive the capsule or if I receive the other formulation, I have the same effect, it's what we need to prove in the viability study. This will be run next year before the phase 3 study. And that's all. I think I do not use too much extra time. Here you have our emails if you have some questions, and you also can do questions in the chat.
That's brilliant. Thank you, Laura. Fantastic to see how much development has gone on with all the work you're doing at Azafaros. And I think there's a community we haven't been involved, from the early stages. It's really exciting to see that. And I know we're really looking forward to the next phases and the next steps as you move forward. And so if anyone has any questions for the Azafaros team, drop them in the chat or reach out to myself, Laura, or Gisela, and we can get those answered. Thank you very much.
Thank you, Daniel.
No, thank you. So I'm going to introduce our next speakers. I'm very excited to say we have two colleagues from Sanofi joining us. We're Isabela Batsu, who's up on stage, and Daisy, who was with us yesterday, who are going to be giving us really interesting talk about innovative trial design and bringing patients' insights to understand meaningful clinical relevance. So I know, Isabela, you're going to be up first. So I'll leave you on the stage and I'll share your slides for you to introduce yourself and to give a presentation. Really appreciate you joining us today.
Thank you so much, Dan. I am very excited to be with you guys today. I know it's these are the meetings that are really very endearing to us because it's all about the patients, and we have you in our forefront, and we really strive to do all the activities and support is with you in mind. So today, with my colleague Daisy, we are going to be presenting some of these innovative trial designs and the study that we're working on bringing patients' insights to understand the meaningful clinical relevance. I am the global project head in clinical development and rare diseases at Sanofi. Next slide, please. So these are our disclaimers. And again, the agent that we will be mentioning is an investigational agent that has not been approved. And just as a brief introduction and as a recap, GM2 Gangliosidosis occurs due to genetic mutations in HEX-A or HEX-B genes. These are rare inherited autosomal recessive disorders, which ultimately leads to progressive neurodegenerative diseases. These genetic variants leads to deficiency for Tay-Sachs in beta-hexosaminidase A enzyme, or for Sandhoff disease in deficiency of beta-hexosaminidase A and B. Deficiency of these enzymes, again, the breakdown of these, that causes the breakdown of these substrates, results to accumulation of GM2. So this accumulation of GM2 occurs primarily within the neurons, and the affected cells become functionally impaired and are progressively destroyed nerve cells in the brain and spinal cord. So this accumulation leads to neurodegenerative symptoms. And again, we could see that muscle weakness, miscoordination, patients losing balance, speech difficulties. So a lot of those different activities that we often see in the patients. Next slide, please. And so this is a cartoon depicting the glycosphingolipid pathway, which is at the heart of multiple rare diseases. Our investigational agent is called Nibaglustat. It's a glucosylceramide synthase inhibitor, and it inhibits this enzymatic conversion from ceramide to glucosylceramide, kind of that first step in that pathway. And it decreases the synthesis of GL1, thereby disrupting the accumulation of downstream glycosphingolipids. So besides all the studies that we have, the amethyst and GM2 gangliosidosis and related diseases, we also have this compound studied in GD3 and Fabry. Next slide, please. And I just wanted to take you on our journey on the evolution of the amethyst trial design at Sanofi. Next slide. So the discussions and the innovative development started around 10 years ago, believe it or not. And we really had very limited data. What we knew at the outset was we knew the disease mechanism, the progression, we knew that there were neurological impacts, and we knew that there was a lack of available treatment. So because of that, there was very limited data on what tools were needed to measure the severity. What was the natural disease course? Was this a fast progressing, slow progressing? How would we develop a clinical trial? And we really wanted to gain insights on both the patients and caregivers' perspective that were living with these late onset GM2 diseases. And in order to develop a clinical study, we really needed to drill down and understand the aspects of how the disease evolves and how fast it evolves to better understand the time span that was needed for us to follow through the patients. And what were the aspects of the disease that impacted the patients the most? Next slide, please. So this was a very interesting endeavor that we partner with the National Tay-Sachs and Allied Diseases NTSAD Foundation, which is kind of the counterpart of CATS in the US. And they have a family annual conference where patients and their caregivers and families come together face-to-face and have different discussions, different forums, different presentations. Well, during those meetings, the patients that were interested to join this natural history study and that consented were assessed by the different neurologists. And there was a battery of these tests that were done looking at severity of gait, motor function, mobility, manual dexterity, speech, doing different questionnaires for quality of life. So all these assessments were done, and they were done sequentially at the family annual conferences from 2015 to 2018. Next slide, please. So from this natural history assessments done at the NTSAD, there were 23 patients that consented that had late onset GM2 gangliosidosis. And you can see the breakdown. 19 patients had with Tay-Sachs disease and 4 patients with Sandhoff disease. The mean patient age was 46, but there was a wide range of patients. So our youngest patient was 24 years of age, and our oldest was 68. The mean follow-up for the natural history study was roughly 2.4 years. Again, that range between 1 to 3 years. And these were some of the disease characteristics from at the first NTSAD meeting. So the age at symptom onset was roughly 21 years. And the age of diagnosis was 31 years. So again, there's this big gap between symptom onset and diagnosis that we had noticed. And half of the patients from this natural history study really needed caregiver assistance. So again, these are assistants to perform the activities of daily living. And around roughly 70% needed some sort of ambulatory assistance, so some sort of supportive device, either wheelchair, walker, knee brace, to help with their ambulation. Next slide, please. So from this natural history, we had gathered and gained very critical insights from the patients and a lot of critical lessons to really understand how to develop an appropriate clinical study. So what we found was that late onset GM2 progresses slowly. So it wouldn't be applicable or appropriate to do a short clinical study. 6 months would not have been enough time to show an impact. And so we needed to make sure that a long-term study was warranted to show this effect on some of the clinical endpoints and some of the measurements that were done. So next is what would be important and relevant to measure? Well, from that natural history study, we saw that there was a slow but significant progression on the 9-hole peg test, this manual dexterity assessment, and the FARS neuro. So because of the natural history and all that work that has been done almost over that time span, 2015 to 2018, we were able to develop our clinical study, and we were able to select our endpoints, neurological endpoints for our Amethyst study. So the primary endpoint you will see, one of the primary endpoints is the 9-hole peg test, and the secondary is the FARS neuro and some of the other ones that have been included in the exploratory endpoints. Next slide, please. So this is our clinical study. The Amethyst study that you may have heard about is in late onset GM2 Gangliosidosis. This is with Nibaglustat. It's a phase 3 pivotal study. It's a randomized double-blind, and this is placebo-controlled study. This is again in adults. We were anticipated roughly 57 patients to be randomized, and they were randomized in a 2-to-1 fashion. That means for every 2 patients that were randomized by chance to Nibaglustat, 1 patient was randomized to placebo. Blinded means everyone does not know if they're getting the actual active drug or placebo. That is a critical component, not to introduce any bias and to be able to ensure that the assessments and the conclusions that you're drawing is based on completely unbiased data. So as you could see from the randomization, we have a 2-year clinical study to be able to determine change over time. At the end of 2 years, all the patients are switched to Nibaglustat open label treatment for an additional 2 years. And during this time points, you could see that there's different assessments being done at baseline and different intervals. There's also a lumbar puncture that is being done at baseline in week 104. That's specifically looking at cerebrospinal fluid and looking at biomarkers, which I will discuss in more details in the subsequent slides. And this is a very innovative clinical study design because, again, usually you just have that top portion with the primary population, the adult, but we have a very interesting design because we included this secondary population, which is a basket trial with ultra-rare conditions within the same biochemical pathway, so related diseases. We also included the late onset juvenile adolescent GM2 patients, GM1 gangliosidosis patients. We had opened it up to supposed C deficiency patients, sialidosis, and juvenile or adult galactosialidosis patients. So I want you to also know a very important factor because for us, and on this side of the collaboration and the support for you is we really need to hear your voices. And when we had designed this study, this secondary population in this basket trial, we only had up to 5 patients that we were anticipating to include. We didn't even know if we could include 5 patients, but there was such persistent and such great feedback from the patient advocacy communities, from patients, from caregivers, that we really understood that there was an interest and really people were hoping to ensure that they're able to be part of the study and to get some opportunities from this. And so we were able to open this up from up to 5 patients to up to 20 patients. And all these patients were on open label Nibaglustat. These patients are on open label for the 2-year primary analysis period and then the additional 2-year follow-up period. Next slide, please. So again, I do want to point out, and this study would not have been possible without all the support from the unbelievable investigators. We had 14 countries, 23 sites, and of course, without the patients that were so dedicated and the caregivers to ensure that, the traveling to the sites and ensuring the patients follow through, because that's a critical component to ensure that we have the data for the broader GM2 community. So overall, we have enrolled 59 patients in the primary population and 16 patients in our secondary population in that basket study. And recruitment was completed in January 2022. So again, with a 2-year follow-up, we do anticipate to have data presented in the first half of next year, which we are excited about. Next slide, please. So just some of the baseline characteristics. I had shared 59 patients in the primary population. The mean age was roughly 37 years of age. And you could see roughly 75% of the patients had adult late onset Tay-Sachs and 25% of the patients had adult late onset Sandhoff. Their age of at onset was roughly 18 years of age, and the age at diagnosis was 29. And again, we could see that very long gap between age of symptom onset and diagnosis. For the secondary population, so the 16 patients that were enrolled in the study, the mean age was 12, and you can see the different distributions between all the different related diseases. So we had 7 patients with juvenile adolescent late onset GM2, 7 GM1 gangliosidosis patient, 1 patient with sialidosis type 1, and 1 juvenile or galactosialidosis patient. So again, we could see that these patients have an earlier onset of symptoms, roughly closer to 5 years of age, and the age of diagnosis is closer to 9. So again, we are very excited about sharing these baseline characteristics and subsequent results first half next year. Next slide, please. And some of the key outcome measures, because again, I had walked you through kind of how we did the natural history study and what we garnered from the patient insights and all the assessments that were done. So we had incorporated the change in the 9-hole peg test for the manual dexterity, and these are co-primary endpoints, as well as the biomarker for the cerebrospinal fluid to understand the change in the biomarker level for those patients. For the secondary population, we are including changes for both plasma and cerebrospinal fluid levels for not only GL1, but disease-specific biomarkers. So for GM1, we're looking at GM1 levels. For galactosidosis, we're looking at GM1, GM3. And of course, secondary endpoints, safety, tolerability, and PK. Next slide. And this is my last slide before I hand it over. So again, this just brings it all together, because as this is an evolution of how the study started and how it evolved and why we picked these specific endpoints. And again, this could not have been possible without the patients and without their insights and without their feedback as well. And the 9-hole peg test was chosen because it's not burdensome for the patient. It's something very easy that can be done. It assesses physical function, motor dexterity up in the upper extremities. It's widely used in other neurological conditions as well. We have that natural history data from with the NTSAD that we saw from the 2015 to the 2018 time period, where we saw a very nice decline over time, so we could actually be able to measure. And my colleague will discuss a little bit further on kind of what is the relevance of measuring this 9-hole peg test in the patients. And last but not least, the co-primary endpoint in the study is the biomarker, the GM2 specific biomarker. And that's why we did the lumbar puncture at baseline and at week 104 to be able to detect the change over time. So as we had mentioned from initially on the first slide, again, there's accumulation because of the deficiency of an enzyme. There's accumulation of a GM2. So we wanted to make sure that if we demonstrate reduction of these accumulation of the substrates with Gangliostat, that would also potentially demonstrate clinical efficacy and, of course, demonstrate that it does get to where it needs to do and pass that blood-brain barrier, being a brain penetrant, to be able to have that reduction in biomarkers. So with that, I will pass it on to my colleague Daisy for the next presentation.
Yeah, thank you, Isabela. Absolutely fascinating again to have the update, the overview of the study, and I'm sure that there'll be many, many questions flying into us. So we will make sure they reach their way to you so we can get those answered. We really appreciate you joining us today to give such an insightful talk. And thank you, Isabela. I'll hand it over now to Daisy. So I'll add your slides so you can click through them yourself. There you go.
Great, thank you. Thanks, Isabela, taking us through the journey of how we make the clinical trial as well as the selection of the endpoints. They are very fascinating because Sanofi spent a lot of time working with the patients, understanding from the patient perspective what are necessary to be included in the trial in order to evaluate a very important drug for them. So this is an exciting time for us to share with our work with you. So as, I'm so sorry, it did not pan out the way I wanted. So to ground us to understand what I'm going to talk about is understanding why is it really relevant to measure the upper dexterity and the lower limb mobility in patients with GM2, GM1 gangliosidosis. So this is a pie chart. It's not like a one-third percentage-wise, but it's to show you the thinking behind the scene, how we conceptualize the relevancy of measuring the patient's mobility. The first part that Isabela demonstrated or explained really clearly to us is that we spend a lot of time understanding the patient, going through the conferences in the NTSAD conferences starting in 2015 over the 4 years' time to understand the patient's experience about their GM1, GM2 experiences. On top of that, we need to take the knowledge from here and bring it on to the understanding if the 9-hole peg test, putting the pegs back into the peg, into the board, is it relevant for these population? And to what extent the upper and the lower motor functioning are important for the patients? Gangliosidosis is going to address these problems. Is it true, right? So that's our question. So what I'm trying to tell you or bring you to our journey is the second part of the story, the relevancy of the 9-hole peg test. So here, what we have done is, from the patient's perspective, we went back to the NTSAD conference and the patient who participated in this family conference. And we included 11 patients of them who attended the conference at the time and tried to understand what does 9-hole peg test really measuring. So from these 9 patients, we want them to perform the test as well as answering some of the questions, some of the other measures at the conference. So give you a perspective of who are these patients. They are 9 patients, a mean age about 49. So it's a wide range as we also observed from the previous presentation, starting with age 36 to 66. 73% of them are male, and majority of our patients are Tay-Sachs disease patients. So we are going to show you what the patient tell us about whether these 9-hole peg tests really measure what they are supposed to measure. But at the same time, we want them to tell us, is it a good measure? How good is good? We say neuro quality of life items, Are they really joining one another together? Are they correlate? Makes sense to them? So that's what we have done. First off, let's take a look at the Neuro Quality of Life item bank, version 1 here. They measure the upper extremity. Now, why we have to pick this is an established scale in the field to measure, like Isabela mentioned about in multiple sclerosis, in ALS. So looking at these items, do they resonate with the patients with GM1, GM2? So here are the actually all the 8 items in this NeuroQual. Turn the key in the lock, brush your teeth, make a phone call using a touch tone keypad, pick up coins from a tabletop, write with pen or pencil, open and close a zipper, wash and dry your body, shampoo your hair. So those are the items. And let's see what they told us, the patients. First of all, the patient told us that 9-hole peg test is indeed relevant to measure the upper extremity function as assessed by the EuroQual upper extremity short form. Now, here are those quotes from the patient who told us how they feel about the 9-hole peg test. They are so relevant. So the first one saying, yes, definitely, the fine motor skills. I've done the 9-hole peg test many times, and sometimes with difficulty. Sometimes with difficulties. I think it's a very good tool. In the middle, the patient told us, I think the 9-hole peg test, good. Because sometimes when I'm putting things in a bowl, it's hard to pick up. Sometimes whatever. So it's hard to pick up sometimes. It's good, because you use both hands and everything for that. So, and reach to actually do it. So this is to give you a perspective that the patient told us 9-hole peg test is indeed relevant to measure the upper extremity. So our next task here is to understand to what extent it really mirrors the activities, the action that they have to perform, right? So all the participants found that the items, we described the 8 items within the NeuroQual actually are relevant to their experiences of the effects of the upper motor skill due to their condition. So they stated that, for example, the Tay-Sachs patient told us, I had a lot of trauma issues too. I did find it hard sometimes, even dialing a phone. Yeah, definitely, it came through. Another patient told us that a few of my fingertips are numb, and I have trouble with fine motor skills a little bit. But like buttons, shorts, skirts, tying shoes, stuff like that, I have more trouble getting the key in the lock than I do turning it. So they give us another perspective. So all in all, you can see that 9-hole peg test is indeed a good measure of the upper extremity for the patients with GM2, GM1 the problem. On the other side, we saw the videos earlier, and also the patient told us about their experiences, upper and also lower mobility function limitation. So from this, we want to understand from a patient perspective, what type of fine motor functions are they experiencing? What type of gross motor function are they experiencing? As we know, the fine motor skill referring to the more coordination of using the precise control measurement, like we were talking about the fingers, the hand, the wrist, and toes like that. For the gross motor functioning, we are relating to the body movement using the larger body parts here, the lower limbs in particular. So we in this particular study, we look at these functions, fine motor skill, gross motor skill from the perspective of the pediatric, highlighted in the blue, and also in the purple are the adult, what they told us about their experiences of having these motor skill at the time of diagnosis, as well as at the time of our conduct of the qualitative interview with them. So you can see that at the time of interview, I will give you a perspective, is that all of these patients told us that they experienced the gross motor function limitation and also the fine motor skill. But at the time of the interview, they told us that basically, yes, they lost quite a lot. The progression is quite severe. So we can take a look at one or two of these quotes from our patients. For example, let's take a look at the time of diagnosis for the fine motor skill from a pediatric patient. The caregiver told us that at 2.5 years, by the time at diagnosis, she was really starting to rake in with picking up food as opposed to using larger sweeps to manipulate food when she was eating, as opposed to using her pincher grasp. She had originally way earlier. She was able to do that pincher grasp. But by the time she reached 2.5 or so, and even before that, really, when we hit the 16 or 17 months, she was raking more. That slowly continued to decline in her ability to actually pick up items appropriately, small items with eating. So at the time of the diagnosis, the adult also talking about typing with all the fingers limitation at the diagnosis. And I would be on the keyboard, and I usually have someone cut my meat for me if I'm having a meal at the dinner. And then when it started to decline, about the age of 27, when the diagnosis comes. So when it's time for the interview, you can see the decline. Let's take a look at the adult who told us how he felt. I do struggle with opening tops of jars and stuff like that. I can open them sometimes, but I do find that often that I do have a hard time opening jars. The bigger the jar, the harder it is to open. So that's the perspective of the changing of the fine motor skill. Along the same line, the gross motor skill set for the patient, the pediatric patient, for example, told us that for the walking, she's easy to fall. Falling is one of the important aspects that they notice right at get-go. And she could not walk. She cannot walk for a long time, yet she's easy to fall. So the symptoms progress from another caregiver of a 13-year-old. She's not able to walk by herself, and she is not able to sit by herself. And looking at the adult at onset, at the diagnosis, at the time of diagnosis, the only thing I noticed for myself, for me, was balance. I had no balance left. I was typically clumsy a lot. Clumsy where things I would normally do, I would trip a lot. I would fall randomly. So you can see the theme here, the balance of fall. And when it progressed at the time of the interview, probably since the time of my diagnosis about 3 years ago, I would fall on my feet, and I probably broke my feet a few times. But about 2 years ago, it was like a year ago. I think it was a lot like 2 years ago. It was probably the worst fall I ever had. When I fell, I broke my foot. I fell, and when I fell. And I could tell I broke my foot. I had a boot from the time before that I was wearing. So you can see the overall theme here. Patient told us, regardless of the age, regardless of the time of diagnosis and interview, falling, using the gross motor skill set, they have limitation. And these limitations progress over time. Importantly is looking at the future. We ask the patients, adult, and also the caregiver to tell us what they think about a treatment, if there is a treatment, that may delay in worsening in the fine motor or the gross motor skill. Is it important to them? All of them tell us that it is very important to them. So we asked them on a scale of 0 to 4. 0 means not important at all, 4 extremely important. How would you rate the importance of a treatment delaying future worsening in your condition, either fine motor skill or gross motor skill? In the interest of time, I will give you one quote from each of the pediatric and also adult patient. For example, the first pediatric patient from a caregiver who mentioned about, yes, a treatment that delay worsening would be important. Still, I think it is helpful because I see the hand change. It bends. Then when you bend your hand for a while, you feel pain. When I think stop bending more and changing the shapes of hands more and cause less pain for him. For example, here on the for the adult, probably a 4 for fine motor skills. Just anything worsening, getting worse is going to make everyday life more challenging with hand strength and grip and coordination with it. And yeah, just the same thing. I would definitely be more interested in the gross over fine since there is where my weaknesses are. If I could do something to stop any of it, fine motor as well, I would still try it. So you can see that patients, they would definitely prefer a treatment that is very important for them if it can delay the worsening of either the fine or the gross motor function. And some other patients prefer more so for the gross motor function. So all in all, I would like to summarize the following for you is that the selection of the 9-hole peg test measuring the upper extremity are very relevant for the patient. And the patient told us it's a relevant tool. And it correlates really nicely with the NeuroQual item bank, as we showed you earlier. And people and patients experiencing GM1, GM2 gangliosidosis experience both the fine and also the gross motor function loss. And these patients reported that a treatment that would delay in worsening for either the fine or the gross motor function is extremely important. So treatment is important for them. Delaying the progression is also very important to them. I thank you for your attention. Back to you, Dan.
No, thank you, Daisy. Absolutely fascinating to see how you go out and get the data, then actually make it usable, especially in a study setting. I know we're going to get loads of questions about this, but in the interest of time, what I'll do is I'll collect all those questions, I'll get them over to you guys, and we can get them answered. But I really appreciate you joining us for the last 2 days to share your experiences.
Yeah,
so
my
thank you very
pleasure.
much, Daisy.
Thank you.
Thank you. So I'm going to move on to our next speaker. I'm really pleased to introduce Dr. Elise Townsend. Elise, how are you?
I'm well, thank you. Thanks for having me.
Thanks for joining us. I know last year we had Mike, your colleague, joined us at the very last minute to give an introduction to this project. And I will let you share how much work has been done in the last 12 months and what you have planned for the future. But I'll share your slides and leave it to you to introduce yourself in a bit more detail and about your talk. So thank you very much.
Wonderful. Thanks, Dan. I'm excited to have the chance to talk with you today about our ongoing project developing an infantile GM2 gangliosidosis clinical rating scale for remote assessment of clinically meaningful health-related functions in children with Tay-Sachs, Sandhoff, and AB variants. I'm Dr. Elise Townsend. I'm both a physical therapist and developmental psychologist by training, and I'm joined by my colleagues. Next slide, please. I'm joined by my colleagues, Dr. Michael Kiefer, who introduced the project last year. He was my PhD trainee and is now taken a faculty position at Virginia Commonwealth University. And Dr. Florian Eichler is our other collaborator. He's a neurologist at Massachusetts General Hospital. Next slide, please. So the overarching purpose of this line of work is for us to develop and validate an infantile onset GM2 rating scale, which is brief to administer, family-friendly, and based on clinical functions most important to caregivers. It's being designed for both in-person and virtual use to allow us to assess children more frequently as they're growing and changing and evolving over time. And the goal is to provide, for this scale, to provide an appropriate endpoint for clinical trials. Next slide, please. So we based the development of our scale and the items in our scale from the natural history of what we know about GM2 gangliosidosis. So much of this is based on retrospective caregiver surveys, which have provided a wide range of functional impairments and neurological symptoms that are associated with the progression of GM2 gangliosidosis. So these are symptoms that you're probably very familiar with. But what we also identified were some gaps. And one is, how can we measure key symptoms and functions prospectively? And how can we do that using a clinical observational rating scale? So that's our first gap. And the second is, we really wanted to come to understand better what symptoms and functions of children are most important to families and to caregivers. And so we designed the study to address these two gaps in our knowledge. Next slide, please. Another foundation for the design of this study is based on FDA draft guidance that provides a roadmap for developing clinical outcome assessments that are fit for purpose and patient-focused. So on the left here, you'll see the sections understanding the disease condition, and then the second in green, conceptualizing clinical benefits and risks. And the early stages of our project, which are now complete, we're really focused on better understanding GM2 gangliosidosis, specifically infantile onset, and functions that can be measured through clinical observation, and then also better understanding the clinical benefits, the caregiver's sense of clinical benefits and risks. So we ask questions like, if your child were getting worse over time, what would that look like? And then more importantly, if your child were receiving treatment and getting better over time, what clinical functions what health-related functions would be important to you and would be meaningful for you to see positive change? And so I'll tell you a bit more about how we measured these things. Next slide, please. So our overall study design had 3 arms or 3 phases. The first and second arm here in green are now complete, and I'm going to tell you a little bit more about what the purpose and what we found in those 2 studies. And then arm 3 is our longitudinal pilot, where now that we have a rating scale, we're going to collect prospective natural history data over time. We're beginning to enroll in that project right now. Next slide, please. So these are the updates from Dr. Kiefer's presentation last year at this meeting. We've completed arms 1 and 2 of the study. Arm 1, we conducted caregiver interviews to gather caregiver perspectives on their children's symptoms and functions. And we did this early in 2023. And what that allowed us to do was to develop scale items, develop items for the rating scale, and also to conceptualize treatment benefit. So to get a sense from caregivers, if your child were improving over time, what would you be seeing and what would you be pleased to see, or what would be helpful? So once we had a collection of scale items that were based on caregiver perspectives, we then took those to a group. We held focus groups and took those to a group of expert clinicians who were occupational therapists, physical therapists, and speech language pathologists. And this allowed us to refine the items in the scale and also to develop scoring criteria so that we could measure change over time in a more granular fashion for each of the scale items that we found. And so that work was completed earlier this year, and we're moving on to phase 3 of the study now. Next slide, please. Could you click it again to see if anything comes through? Oh dear. Okay. I'm not sure what happened, but I can explain to you what I intended to be on this slide.
Give me 1 second. Let me just put them in as a PDF. It may sort of formatting up. 1 second, but carry on talking.
Okay. So a little bit about our results. So what we have now, based on caregiver interviews and expert clinician focus groups, is we have a 16-item scale of health-related function for infantile onset GM2. And those 16 items are divided into 5 different functional domains. So when we asked caregivers what was important to them, they talked about communication of their child, the ability of them to develop sounds and make sounds, their nonverbal communication, and also their ability to understand communication. So that was domain number 1. The second domain was the feeding domain. So feeding behaviors and their ability to take food and drink orally was also important to caregivers. The third domain was the visual domain. So can the child use their eyes to track objects in the horizontal plane? And can they move their head and their eyes, for example, to follow a moving person across the room? The fourth domain that came through in our study was hand and arm use. So the child's ability to reach and grasp and manipulate objects. And the last was the gross motor domain. So things like their ability to control their head, their ability to sit, their ability to be on their belly and move forward, and their ability to stand and walk. Oh, fantastic. You got it back up. Okay, good. So these 5 domains and 16 items were really the results from the first 2 phases of our study. And this comprises the scale that we're now going to take forward and collect longitudinal natural history data for. Next slide, please. So the red box circles the piloting of the rating scale, which is the section of the study that we're beginning right now. And so what we're doing now is recruiting 20 or more children with GM2 Gangliosidosis and collecting that prospective natural history data related to their health-related functions. So how are these different functional domains? How are the children functioning? And how is that functioning changing over time? Next slide, please. So who can participate in the study? We're recruiting children under 5 years of age who have infantile onset GM2, so Tay-Sachs, Sandhoff, or AB variant, and their caregivers. Participation will involve 30-minute Zoom visits every 45 to 90 days. So we're sampling more frequently in the younger children who we expect their function will be changing more quickly over time, and less frequently in the older children who we expect will be a bit more stable. Participation will also involve online surveys that are pretty reasonable in time and scope, so that we can also continue to gather the caregiver's perspective of their children's symptoms and function. Next slide, please. We especially want to thank the National Tay-Sachs and Allied Diseases Association for funding this work. The work would really not have been possible without their funding. It funded Dr. Kiefer's dissertation time. And we also want to extend our gratitude to the 10 caregivers in 8 families who participated in Arm 1 of our study, as well as the expert clinicians who are really a global group of clinicians with expertise in pediatric progressive rare diseases, including GM2, who helped us refine the items of the scale and develop scoring criteria. So if you'd like to, if you have a child who meets the inclusion criteria for the study, and if English is a language in which you can communicate, then we can recruit your child for the study. Feel free to use the QR code below or contact us by email if you prefer. And if you want to learn more about the study, we can share more information. Thank you very much, Dan.
That's fantastic. Thank you, Elise. I know there's going to be lots of interest. So what I'll do is when we do a summary of the event, we'll make sure all the links are shared, direct people directly to you for any questions. And hopefully next year you can come back and give us another update, because I think it's been great to see, you know, over the last couple of years, the work that's been going on and what you've got planned for the future. So thank you again for joining us today.
My pleasure. Thank you.
Thank you. So I'm very excited to welcome our next speaker. So this is Hannah Scott. So Hannah Scott's going to talk to us about a project called the C-POS, which is the Children's Palliative Care Outcome Scale, which is being done by Hannah and her team in the UK. So it's something actually that my wife and I have taken part in. I really want to get everyone to listen to this talk. I think it's going to be absolutely fascinating. So Hannah, I'm going to hand it over to you to introduce yourself in a bit more detail, and I'll just share your slides. So thank you.
Thanks, Dan. Hiya, I'm Hannah, and I'm a research assistant and PhD candidate, and I work on the Children's Palliative Care Outcome Scale study, and the principal investigator who's leading the study is Professor Richard Harding. And we're funded by the European Research Council and supported by NIHR So C-POS started because my colleague Lucy Coombs, who's a clinical nurse specialist in the UK, conducted this systematic review, and she found that there isn't currently an ideal outcome assessment tool for use in paediatric palliative care or with children with all life-limiting, life-threatening conditions outside of sub-Saharan Africa. Generic health-related quality of life measures that exist aren't relevant to children with palliative care needs, and in disease-specific measures, they're only relevant for that specific population. So we know from adult practice that outcome measures improve service quality and promote patient-centered care. They lead to better symptom recognition, increased referrals, and overall lead to better quality of life. And developing an outcome measure for all children with any life-limiting or life-threatening condition has been a research and clinical priority for quite a few years. So we're hoping that through developing C-POS, it will help children and families to identify priorities and outcomes of care with clinicians. It will help clinicians to conduct more thorough assessments and monitor outcomes over time. And it will help commissioners who are the ones that choose which services get funded and decide how much funding goes to different services, ensure that actually services are patient-centered and outcomes-based, and that they can actually see and measure the benefit the services are having. So the overall aim of the C-POS study is to develop a child and family-centered outcome measure that can be used by all children and young people and their families affected by any life-limiting or life-threatening condition and test its psychometric properties. And the study has gone over 3 phases: development, validation, and implementation. So we started with the development, and we started with a really large interview study where we interviewed 106 participants, including 26 children and young people with life-limiting conditions, 40 parents and carers, 13 siblings, 12 commissioners, and 15 health and social care professionals. And what we found when we analysed that data was that the symptoms and concerns that were priorities for children and their families mapped to the World Health Organization domains of palliative care. So the physical, spiritual, existential, psycho-emotional, and social and practical needs. But for this population in particular, there was this overarching theme of normality as well, which spanned all the domains around children and their families not knowing any different because children had been born with a condition, or children, particularly in the cancer population, where they'd had cancer and they'd gone through treatment and were now trying to return to their normality, or for children that were diagnosed later on in life, adjusting to a new normal, and families trying to adjust to that new normal with diagnosis. So we had all these potential symptoms and concerns that could be included in a measure, and we needed to work out which ones to include. But we also needed to work out how to actually design the measure. So my colleague Lucy again led a systematic review, and she looked at the evidence that existed on recall period, response formats, and modes of administration. So how could we actually enable children to be able to participate in these measures as well? And from that review, there were 8 recommendations, and that included things like short recall periods, where children aged about 5 to 7 generally can't remember back reliably for more than 48 hours, making sure measures are visually appealing, and having different versions for different age groups or cognitive abilities so that children can participate in a version that's most appropriate for them. We then took all of that information on what we should include in a measure and how we should design it, and did what's called consensus work. So we did a Delphi survey where we asked healthcare professionals and parents to rank which items were most important, and we came up with a top 10 list. We then went to a young person's advisory group and asked the young people what their top items were. And then we held an item generation meeting with our expert stakeholder group, and we ended up developing some initial versions of the measures. And these were our first versions. We had 2 versions for parent proxy reporting, so one for children of under 2 years and one for children of 2 to 17 parent proxy, and then 3 self-report versions, so children 5 to 7 years old, children 8 to 12 years old, and children 13 to 17 years old, or cognitive equivalent. And we worked with the young people's group to name these in a way that wasn't based on ages, and the young people's group advised us that planets were the best way to do this. So we have 3 versions for children to self-report: Mercury, Saturn, and Neptune. And we ended up covering all of the 4 domains again in both our child self-report items and in our parent carer items that we included in the first versions of these measures. So the measures cover pain and other symptoms, being able to ask questions, being able to undertake usual activities, worry, sharing feelings, being able to do things you enjoy, and living life to the fullest. And for parents and carers, getting enough sleep, having access to information about the child's condition, support needed to care for the child, support to plan for future care, and support and the impact of the child's condition on the family. And we then took that into the validation stage of the study. So we wanted to test C-POS to make sure that families understood the questions and work out the best recall and response formats for them. So we cognitively tested using Think Aloud methodology, where we asked participants to read the C-POS questions and then explain their understanding of the questions in real time. And we then tested recall periods of the past week and yesterday and today, and a response format of a frequent scale and a 5-point scale, and used emojis with children to anchor the scales. And these cognitive interviews were really helpful because they helped us to make changes to the versions, particularly to be inclusive of children and young people who are nonverbal. We've now taken these versions that have gone through a number of revisions through cognitive interviewing into the psychometric testing, which is essentially testing on a much larger scale. So we're looking to see, does C-POS measure what it's intended to measure? Is it doing so accurately and consistently? And can it detect change over time? And we're currently working with over 60 sites across the UK who are recruiting children and families to our studies so that we can test this. Whilst the large-scale validation is ongoing, I've been leading on the implementation work as part of my PhD, because there's no point in having done all this work to develop a measure if we then can't get it into practice. So this started with a systematic review to look at how can we implement outcome measures in paediatric healthcare generally, what's already out there. And we identified barriers, including lack of knowledge about how measures might improve care and outcomes, the complexity of using and implementing these measures, staff readiness and willing to change current practice, and potential disruption to workflows, and lack of resources to enable implementation and sustained use. But we also found facilitators for implementation. So educating and training staff and families on how to implement and use outcome measures and the advantages of using measures over current practice and the benefits they had on patient care and outcomes was a really helpful way of ensuring these measures got used. Addressing logistical and resource barriers like time, funding, staffing help enable measures to be used. Having a comprehensive plan for implementation and formally appointed implementation leads or champions can also help ensure that measures get used. And importantly, having buy-in from staff and committed support from leadership in organisations is really important to making sure that outcome measures can be implemented successfully. We then went into use some of the data from the qualitative interviews that I spoke about earlier to look at what different stakeholder groups thought about the barriers and facilitators or the challenges and benefits of outcome measures, specifically in paediatric palliative care for children with life-limiting conditions. And I'm currently still analysing some of this data. And then, as Dan mentioned, we did some workshops with parents and carers and healthcare professionals and bereaved parents to work out a plan, essentially, for how we actually go from all these potential strategies that may or may not work from paediatrics generally, and what we do know about the palliative care and life-limiting, life-threatening care setting, to actually having this measure and getting it into practice. And we developed a map and a provisional implementation plan for that. From that, our next steps are to continue working on our large-scale validation with the hope that once we have it validated, we can start actually using this plan to implement C-POS into practice. Once C-POS is validated, because it's part of the POS family of measures, it will be available through the POS website, and you can register interest on the POS website at the moment as well, so that you can hear as soon as C-POS is ready. And we're also working with a number of international collaborators. So while we're doing validation and implementation work in the UK, our colleague Dr. Ivna Masango is leading on implementation work of the African version as part of the GAP study. We also have colleagues that have been doing a similar study in Turkey and Jordan, particularly for refugee children. We had a MSc student recently do some cognitive testing of the UK version in Singapore, and have colleagues that have joined us from South Korea and Japan to translate and cognitively test the new versions of the C-POS for use in South Korea and Japan, as well as colleagues in Australia and New Zealand that are joining the psychometric validation study to be part of that as well. I just want to thank our funders, as we couldn't do this research without them, as well as our steering group and our PPI members, as well as the young people's advisory groups we work with at Great Ormond Street Hospital and Leeds Children's Hospital.
That's brilliant. Thanks, Hannah. Absolutely fascinating project. I mean, when we first found out about it, I thought it was really brilliant and really going to unlock a lot of insights. And I hope in next year you come back and give another update of the work that you've been doing.
We'd love to.
Yeah, I'm really excited. I really appreciate you taking the time to present today. So thank you very much.
Thanks, Dan.
Thank you. So now I'm going to introduce our final speaker, who's a familiar face to many of us. It's Kathy from the NTSAD. So Kathy, you can introduce yourself. I'll get the slides up that you wanted to share to talk through the Global Alliance, but please, I'll hand it over to you.
Sure. Hi, everyone. Thank you so much, Dan, for the opportunity to share about the Global GM1 and GM2 Alliance. It is certainly a pleasure to join you. As Dan mentioned, my name is Kathy Flynn, and I am the CEO of the National Tay-Sachs and Allied Diseases Association, or NTSAD, as we are commonly called. Our organization was founded 66 years ago, and we're based in the United States, but we often support families abroad who reach out to us as well. Some of you may have met my colleague, Deanna. She's our Director of Family Services at NTSAD. I believe she spoke at last year's European Family Conference, if I'm not mistaken. We can go to the next slide, Dan. So as stated in our mission, NTSAD leads the worldwide fight to treat and cure Tay-Sachs, Canavan, GM1, and Sandhoff diseases by driving research, forging collaboration, and fostering community. And we do that by building community, connecting families, accelerating research, and providing much needed support to the families who have a loved one with one of these rare disorders. I can honestly say that supporting families is truly the center of everything that we do. Next slide, please. The CATS Foundation, under Dan's leadership, and NTSAD have been working together to launch a new international initiative called the Global GM1 and GM2 Alliance. We saw value in convening a group of advocacy leaders from around the world to discuss issues that relate to and impact both the GM1 and the GM2 Tay-Sachs and Sandhoff communities. So as drug development for these rare diseases advances to preclinical studies and clinical trials, we believe that there's an opportunity to create this global coalition and strengthen the worldwide GM1 and GM2 community. Our goal of the Alliance is to unite, to empower, and to advocate for our global community. And establishing this group will help us build those strong connections, allowing us to share information and collaborate not only across the advocacy groups, but also with industry on a global level. Most importantly, and in summation, it really is bringing us together to amplify our collective voice worldwide. On the right side of this slide, you will see a list of 11 advocacy groups representing several countries who attended our first meeting last month, as well as 4 industry partners who have expressed support of this initiative. And we really do hope this will grow. Next slide. Currently, we are in the process of formalizing the GGGA and surveying those who attended the meeting to determine where the greatest needs lie. So what are our families telling us? What do they need? And what can we do collectively? We will use the feedback from the survey to create a roadmap for projects that the Alliance can address. After we are established, our hope is to support emerging groups in countries or regions of the world where they do not have a dedicated group and there is a need for support. So if you serve the GM2 community or the GM1 community and are interested in learning more about the Alliance and possibly joining, please reach out to Dan or to me. My email address is listed here on the slide, and I recall Dan mentioning that he will also be sharing contact links. But we would love to hear from you. We would love to have you engaged. Next slide, please. I'd also like to take the opportunity today to let you know about an important meeting that NTSAD is hosting with the U.S. Food and Drug Administration. And for those of you in Europe, you may be more familiar with the EMA. The FDA is the U.S. equivalent of the EMA in Europe. On February 15th, NTSAD will be hosting what is called an externally led patient-focused drug development meeting. It is a 5.5-hour virtual meeting dedicated to the topic of GM2, Tay-Sachs and Sandhoff diseases. The meeting will include clinical overviews of the 3 onsets of GM2, infantile, juvenile, and late onset, and will feature several personal statements by individuals, family members, and caregivers impacted by GM2. The purpose of the meeting is to really help the members of the FDA learn from the experts, and the experts are the patients and the families, to help the FDA best understand the patient experiences, the family experiences, the perspectives, the needs, and priorities, especially when the time comes for them to evaluate and approve drug development and clinical trial designs for GM2. The content of the meeting will be captured into what's called a Voice of the Patient report for reference after the meeting as well, and we'll be happy to share that with those who may not be able to attend. Our goal is to have as many members of the GM2 community attend this meeting so that the FDA has a full understanding of the worldwide impact that GM2 has on individuals and families, and also our collective desire to accelerate research for therapeutic developments. It will be an interactive meeting with opportunities for the audience to participate in polling and either phoning in or writing in real time. And we're currently in the process of creating a dedicated web page for this meeting. And we will be encouraging people to register and share their comments and questions directly on that registration page. And that registration page will also be open after the meeting if you think of anything else that you might like to share, or if you weren't able to make the meeting but would like to be part of that Voice of the Patient report. So as soon as that link is available, we will be sharing it with members of the GGGA, and we will also ask Dan to share it with today's conference attendees. Let's go to the next slide. Individuals and families who participate in this GM2 PFDD meeting with the FDA will make the greatest impression on those FDA decision makers. But there will be other key attendees invited to attend, and they include patient advocacy organization leaders, those in the research community, clinicians, and industry partners. This meeting is open to the general public, so we are hoping to have a very large turnout. To that end, I personally invite everyone attending today's conference to register and join us at this meeting. And I encourage you to invite others to attend as well. So if you are a patient, family member, a researcher, clinician, or industry partner, we really do hope to see you at the GM2 PFDD meeting. Next slide, Dan. NTSAD has hosted other interactions with the US FDA in the past. Specifically, we hosted 2 listening sessions. Listening sessions are much shorter in nature. They are 90-minute meetings, and they are closed, meaning they're only open to the attendees who are speaking and the members of the FDA in attendance. In January of 2021, we hosted a listening session focused on late onset GM2. And in March 2023, we hosted a listening session on Canavan disease, which is one of the diseases NTSAD also supports. During these sessions, patient families shared their very personal stories of diagnosis and symptom management, as well as their hopes for approved treatments. Unlike the listening sessions, the GM2 patient-focused drug development meeting is a much longer meeting, and it's open to anyone who wishes to attend. And perhaps the most impactful thing is that it also involves audience interaction, and so your voice can be heard individually and collectively. So in closing, I do invite you to save the date, the 15th of February, mark it on your calendar and watch your email inbox or your social media feed for a link to register. And last slide, Dan. Just thank you for the opportunity to join you all today at the 10th European Tay-Sachs and Sandhoff Disease Conference, and to be able to share information about the global GM1 and GM2 Alliance, as well as the GM2 FDA meeting. My contact information is available on the slide and will also be shared, so please certainly reach out if you would like more information. Dan, back to you. Thank you so much.
No, thanks, Kathy. I think it really shows 2024 has the potential to be a really big year for the community with the establishment of our global alliance, which I know we both believe is going to make a big difference, but obviously with the PFDD as well. So much is happening that I'm really excited in the next 12 months to see where we get to. And I really appreciate you taking the time today just to give everyone those updates, and we'll, of course, share everything out to everyone.
My pleasure. Thank you.
Thank you, Kathy. Now we're going to welcome back Bea. Hi, Bea, who's going to give us a quick review of the day, just to share maybe some insights that Bea's put together from all the interesting talks we had. Over to you, Bea.
Thank you, Dan. Well, thank you everyone who has participated in a very interesting day. Well, I'm going to sum up what we have been talking about to make a summary about everything. Dan started talking about the important role of the patient organizations in the puzzle of the rare disease world, where we play an important role working together to make sure that the patient voice is heard, to keep the patient view during all the clinical and research process. It is important here to point out that patient organizations are not responsible for scientific research, but we try to be the patient voice in those processes. Then we had Taylor Field, who very kindly recorded her interview from Intrabio, a summary of phase 2 findings for GM2 and Ng-Mak with Nibaglustat at Intrabio. It is important to mark that these results are referred to trials with adult population, and that they are working on to the next steps for next year to broaden that scope. Then we have Mathias Schmidt from JCR, a pharma company that research on lysosomal storage disease. They are working in a new molecule that could offer some possibilities for this type of storage disorders. Dr. Mengel, CEO as Spin CX from Germany. He has joined again us in a new edition of our conference. He has presented a detailed work about the clinical side of GM2 diseases and other similar diseases too. Its progression at a wide range of age, and they have enrolled 65 patients, I think, all over the years on an 8-in-1 study, and he has presented detail of his natural study of the development of the symptoms from all this range of patients. I want to say here a very huge thanks to our college, Hand in Hand Charity at Germany, to Birgit and Volker especially, because the video they have presented to sum up this work has been really very nice. I have enjoyed a lot to see this video. That has show families, which is the most important thing of this conference. It has been very nice as part of what we try to do here is bring families together in a way in this virtual conference. This video has achieved this goal. So thanks for the team at Hand in Hand. It has been beautiful. Then we had Dr. Laura Lopez de Frutos from Azafaros, a company mostly in Europe working on a new compound, Nibaglustat. They are carrying on a Pronto study, which is a prospective longitudinal study of neurological disease trajectory in children living with late infantile or juvenile onset of GM1 and also GM2 Gangliosidosis, that has the goal of being developed in 6 countries with I guess 12 sites in a timeline from 2022 to 27. Currently, they have enrolled 16 patients in 4 countries. They have an interesting path ahead, not only studying the natural development of the disease, but also testing the safety and efficacy of the new compound. Isabela Batsu was next speaker. She's Global Project Head Clinical Development at Rare Disease at Sanofi. And she has gave a talk about innovative trial design, an approach that has brought patient insights to understand meaningful clinical relevance. For this, she has talked through the very valuable work that Sanofi has completed in relation to the natural study of GM2 starting back many years ago and mainly focused on late onset patients. Together with her, Dr. Ng-Mak, who is GM2 HEVA business partner of Sanofi, has talked about the results and its relevance on the study in the patients that and difference perspective about those results. Then we have Elise Townsend, who is an associate professor of physical therapy at NGH Institute of Health. She has talked, we have her partner last year, and he has talked about this study design that developed an FDA draft guidance, but most of all, many interesting data about how to understand the disease. They have conceptualized clinical benefits and risks and has selected the outcome measure. Very interesting and complete work too. And then we have had Hannah Scott from King's College London, also with a lot of details from, she has introduced the children's palliative care outcome scales they are working on at this moment. It's a very interesting work about different versions from children and parents, testing different groups of affected population to implement in the care for children with life-limiting condition. And finally, our colleague Kathleen Flynn, CEO at National Tay-Sachs Analysis Organization at the US, she has gave an overlook on their work, and mostly what is, I think, is very interesting for us, she has explained the project concerning the global GN2 and GN1 alliance, the opportunity that we have to create a global community for all the patient organizations to work together worldwide, and that we have already started to work to make it come true for in the months to come. So this is a very exciting project that we have ahead. So thanks to everyone for joining us today in our 10th European Conference. I think, Daniel, that this has been a very interesting 2 days with a lot of information, I guess, but to have an interesting overview of how are things going on in our GM2 community. Thanks everyone that has connected with us, and thanks especially everyone to all of the speakers. And thank you, Daniel, for putting everything together from the Tay-Sachs, from the CATS Foundation.
That's fantastic. I just want to say a big thank you to all the speakers and attendees for another great event. Any feedback, please just send it on to us, and we will share the recordings from today if there are sessions you want to rewatch or hear some more information about. And we look forward to potentially seeing you all in person at some point next year. So thank you once again, and enjoy the rest of your day. Thanks, everyone.
Thanks, everyone. See you next year.
Bye.